This ugly multi-allelic gene family is presenting super hot antigens, and basically, you are flipping stupid. How? Just watch the free stream. I am unreasonably proud of this meme. I can't wait to talk to you guys today about my favorite protein ever. Yeah, that's what we're doing today. But before we get into it, let's say hello. Hello, Level Up. Hello, King. Hello, Hero Blaze. Hello, Tritron. Hello, Clockwork. Hello, Hanny. Hello, Caribbean. Hello, Will Not. Hello, Archie. Hello, Ellie Dewey. Hello, Pathogenic. Hello, Ryu. Who else we got here? Hello, Clockwork. Hello, Vietran. Hello, Marlon. Welcome, welcome. Hello, Guy. Welcome, welcome. Hello, Corvid. Hello, Dreamless. Hello, Isatan. Hey, Toxo. Welcome, welcome. Yeah, all right. And hi, Marshy. Hello, hello. hello. Yes. My favorite protein here today. Yeah, not a lot of pre-chat today. I just want to freaking get into it. I'm really excited. And to all of you who are studying for exams right now, I wish you all good luck. It is about that season, aren't we? All right, let's freaking get into it, why don't we? So, what is the protein that we are going to be talking about today? Oh no, do we have the stream chat is stuck again? Why does this happen? I don't understand. Hold on. Let's refresh that. Okay, there we go. That should be good now. Hopefully that will update live. Just let me know if it's still funky like that. Today, talking about my favorite protein, all right? What is my favorite protein? My favorite protein is MHC class 1s, which stand for Major Histocompatibility Complex 1. It is a protein that is essentially a very centralizing force in the antiviral meta. It's the whole reason why I got into this weird space of immunology to begin with, and I want to tell you a little bit about it today. All right. MHC2 stands for the chat. Nah. They're barely going to get a mention today. All right, so what is Major Histocompatibility Complex 1? So, again, this is a centralizing force in the immune meta. Thank you so much for the super chat, Will Not. Thank you. Good luck on your biochem exam. Good luck. Good luck. I'm sure you won't need it, but good luck anyway. All right. So, what does MHC do? How does it function? Why is it so centralizing? So basically, these proteins are a class of proteins that sit on the cell membrane and display digested protein fragments to T cells. T cells being a class of adaptive immune cell that can do things like regulate the immune system, but also in terms of the CD8 positive cytotoxic T cells, directly do some cell killing of their own. And so MHC kind of acts as this cell health sentinel. If it displays an antigen that belongs to you, a cellular protein, T cells are going to ignore that. But if it displays an antigen that is not native to you, that's when T cells get kicked into high gear and start doing a little bit of cellular murder. So I love this protein. And why is that? Because myself, my taste in stuff that I enjoy tends to be, let's say, eccentric. I think of MHC class 1s as being a highly eccentric class of proteins, and there's going to be all sorts of little weird niche interactions this protein has, which I absolutely love. All right. As Chad has said, these proteins are not only important in terms of detecting whether or not a cell is infected, but also cancers will interface with this protein. There's a whole bunch of interesting immunology, embryology, evolutionary biology, all centered around this centralizing meta, which I think is super cool. All right. So the first thing to know, the first headache to get past, is this understanding that the family of MHCs is unsurprisingly vast and diverse for being an immune protein, right? This should not come to a surprise because in terms of the fronts of evolutionary innovation, we'll say, there is, in my opinion, no field more bubbling with new evolution than immunology, right? Viruses and bacteria and fungi, these are all entities that are constantly pushing our immune systems to co-evolve, right? So it should serve no surprise that the proteins of the immune system are also going to be similarly varied and diverse. So here I have the 3 classes of MHC. We're going to be focusing on just class 1 for today, but just to give you an overall idea of what these other guys do, I have them laid out here side by side, right? All right. So, class 1s, which is what we're going to be talking about today. So these are super diverse. That means you have quite a bit of them, as well as the fact that across different primate species and mammals in general and vertebrates in general, they're going to have a lot of funky individualism with regards to what MHCs they express. Thank you so much for the super chat, Pathogenic. You generally saved my life by doing a video on immunology. Is my exam tomorrow? Thank you so much. I'm not entirely sure how useful this PowerPoint will be. I just hope this is something that keeps you invested and interested in immunology, but I actually don't know how much of this is going to be directly relevant for your exam tomorrow. But thank you so much for the super chat. MHC2 are the professionals, MHC1s are the amateurs. What's with this? What's with this hate, y'all? We can all come together and agree that MHC as a family are quite excellent and interesting. But yeah, so anyway, moving on. Class 1s are very diverse across species, across individuals. They're basically, you could count them as your immune fingerprint, right? They're very individualized and highly variable. And the job of the class 1 MHCs is to present internal antigens. So what's inside the cell, we're going to display that to the outside. And almost every cell in your body will make this and display this. So that's all nucleated cells. So any cell with a nucleus will be making and expressing MHC1. All right. So what about class 2? So class 2 is slightly different, but very similar in function. So these are more conserved, meaning that there's less individual variety between species, between individuals, and the job of the class 2s are to present external antigens. So really the only cells that make an appreciable amount of class 2 are the professional phagocytes, like macrophages, for example, or dendritic cells, right? These guys are cells who have been optimized to eat things in their environment, and then they have a pathway called the cross-presentation pathway where they can take proteins that are on the outside of cells and display that for T cells to ponder, we'll say, to ponder. So then we have class 3, which are the most mysterious members. I actually don't know a whole lot about the class 3 MHCs. There's not really a consistent theme with these guys. They're named class 3 because they were found in between the class 1 and class 2 genes on the chromosome. But they're generally immune system stuff like complement, pathway stuff, heat shock stuff, cell signaling stuff, just kind of randomly associated with these other genes. Partly immune in theming. There's a question in chat asking whether or not heat shock stuff is immune. I mean, again, with biology and naming and the fact that words are just 8 noises that have finite meaning to express infinitely complex ideas, heat shock is more of a stress response, but immune responses are also a stress response, so there's some commonality there. But it's not what I would classically consider to be the immune response. Although I will say heat shock and protein misfolding, I'm less familiar with that and how that plays into immunology, but they are both stress responses for sure. So with this in mind, we have to accept that these 3 boxes describe an incredibly vast array of genes and proteins, and the ones that we're going to be talking about today are the class 1s. Class 2, the certified glutton class, affectionate. Yeah. Exactly. Okay, before we can appreciate how cool MHC class 1s are, I first have to tell you how they function. And to do that, we're going to walk through the antigen presentation pathway that the class 1s use. And this is going to be a very simplified version of this walkthrough, but I think in order to appreciate how clever the cell can be, we should talk about how this functions on a cellular and molecular level. So, MHC class 1s are a family of proteins whose job it is to display an array of internal proteins, both cellular and foreign, right? So, if your cell is healthy, then your MHCs should only be expressing antigens, that is little fragments of proteins that are from you, right? But if they're not from you, if these peptides are foreign, that's going to cause problems. That's going to activate the T cells. So if we're talking about a viral infection, we first have to talk about where these proteins come from. And these proteins come from the fact that when you are infected, I have the flu virus here. When you are infected by the flu virus will enter the lung epithelial cells. And so in the process of just existing inside the cells, some of those proteins are going to be chopped up just by chance, right? So there's this protein complex called the proteasome. This is like a garbage disposal for proteins. Right, so there's this barrel with a bunch of angry, chemically active protein residues on the center of this barrel, and essentially you can imagine this as just basically a vortex of blades. So flu proteins enter from the top, and then as they move through this garbage disposal, they will be chopped up into peptides, right? Peptides, aka fragments of proteins. Yeah, Marlon says paper shredder, exactly. Garbage, wood chipper, garbage disposal, paper shredder, whatever you want to call it. The proteasome is no joke, right? The proteasome is regularly involved with protein recycling. So the proteasome doesn't know that a protein is viral or not. It's just chewing up whatever proteins have been sent to be recycled. Okay, so that's going to include tiny little peptides from your cells, so cellular proteins, as well as viral proteins. So that's why we get these various different peptides, typically between 8 and 11 amino acids long. Will the presented proteins ever degrade so that new proteins can be presented? That's an excellent question, Dreamless. So MHC is recycled. So you just pull the entire MHC out from the membrane and recycle the entire thing. That'll get rid of that old antigen so that new MHCs can replenish the surface of the cells. So the next step is to then take those peptides, remember those chewed up pieces of proteins, and transport them into the endoplasmic reticulum, or the ER as we call it. So this is done through a protein called transporter, whoops, bit of a typo there. Peptides bind to the transporter associated with antigen presentation, aka TAP1. This is a membrane channel that allows for small peptides to flow through from the cytosol, aka the inside of the cell, into the organelle called the endoplasmic reticulum. And the important thing to remember about the endoplasmic reticulum is that it is a hub for protein processing. And if you don't remember exactly what the endoplasmic reticulum is, don't worry, I got you. So let's see if is my mouse showing up on screen? No? Hello, mouse. Okay, there we go. Okay, so here we have the nucleus, cell nucleus, and here we have an assortment of endoplasmic reticulum that are connected to the nucleus. And so, I'm a little cut off. Hold on one sec. Let me make sure that I'm not. There we go. Much better. Okay. For some reason I wasn't being tracked properly. Let's get myself recentered. There we go. Where was I? Mouse on the screen. Okay, yeah, so the nucleus, this is where your DNA is. It's where your mRNA is being made and shuttled off to the ribosomes, which all occurs out here, the outside of the cell. And the endoplasmic reticulum is these multi-folded, multi-layered batch of membranes that are studded with ribosomes. So this is where a lot of your proteins do get made, especially if their destiny is to go out into the membrane. And so MHC, being a protein that is that's technically not the right word, displaying peptides to the outside of the cell, MHC is going to have a history of being assembled within these cellular compartments, okay? So how does MHC actually get the protein that is being displayed? Well, TAP1 enables short peptides to cross the barrier between the outside of the cell, not the outside, but the inside of the cell, like say over here. And then TAP1 transporter will enable those antigens to flow into the endoplasmic reticulum where MHC actually lives. And then there's another loading process by which MHC gets loaded in with that peptide. That whole thing gets shuttled off. Pretty sure it gets... cell biology brain fart. I'm pretty sure it gets shuttled off to the Golgi, where it then becomes destined to leave and go and be attached to the cell membrane. Okay. This is also why you have to learn the boring stuff about the cell too, right? So if you're in intro bio and you are so bored of hearing about the Golgi and the ER, I was right there with you when I first started learning about it. But then when you apply basic cell biology, basic organelle biology to specific problems, these get a lot more interesting. Okay. I will stop for questions at some point after I finish this whole pathway. So, don't worry if this seems a little confusing. All right. Protein logistics are so important, even if so every day. Yeah, exactly. Like, I think when you learn about a lot of these basic concepts like organelles and stuff, on first pass, it can get kind of boring. I think where it gets almost universally interesting is when things go wrong. I think that's when things get very fun. And I think, I can't really speak to American education these days because public school teachers are so overworked anyway, that just asking a public school teacher to do more just feels so bad. But I think if you wanted people to be interested in biology, you not only have to teach when biology goes correctly, but also occasionally give them stories about what happens when biology goes wrong. I think that's the best way to present this kind of information. Nothing boring about it. Wait till they learn about scrambled proteins that delineate the ER from the Golgi. I've been a long time hater of the Golgi on my channel. I don't want to, I want to be very honest with my audience, right? That way that like, if I do find something cool about the Golgi and I do a heel turn, I'm like, actually the Golgi is pretty cool. I will let you all know. Because at this, on one point, like, I think my job is to be a cheerleader for all of biology. But on the other hand, like, I feel a duty to be honest. And maybe it was some kind of trauma from grad school where I like didn't get the Golgi, how it really functioned. And then that put a distaste in my mouth. But I will take your word, Clockwork, that there's something cool about that. But I don't actually know exactly what you're referring to. Hello again, Phi. Hey, Reunite. Welcome. Oh, the proteins are called scramblase and flippase. Oh, I see. Wait, is that for delineating ER from Golgi? I thought that was just to flip lipids inside and out. Yeah, I don't remember. The Nero poster hates the Golgi. I know. I'm also an immune cell, right? The Golgi is incredibly important for macrophages and other phagocytes like neutrophils too. But I just, when I was learning about the Golgi, I just couldn't. I just couldn't. Okay, so once you get those peptides, those fragments of proteins that have been digested either from cells or from evildoers, pathogens, bacteria, viruses, once you get that into the ER, there are other proteins that help load that antigen into the groove of MHC. So over here you can see I have the blender render of MHC class 1. And in blue we have the actual MHC protein. That's the equivalent of this cartoon over here. And in magenta we have a peptide fragment, which I've kind of scribbled up here in the cartoon version as well, right? So in the lumen, so the inside of the endoplasmic reticulum, chewed up peptides bind the antigen binding groove of MHC. So that's, and they do that with the help of other proteins that I'm not really going to mention here. Then once you have successful loading, you are going to shuttle off MHC into the Golgi, and then from the Golgi, they will leave the Golgi and head towards the lipid membrane. So this circle, this lazy circle of mine, is an example of the lipid membrane, and then that will merge with the outside, and that's how you get entirety of transport from beginning to end. That's MHC's story here. So here is a summary of what I just said, and I'll pause here for questions, comments about how MHC class 1 functions, what its job is. So I'll just repeat it here. First, a virus. Again, I'm only referring to viruses here for the sake of simplicity, but this story starts off with a virus infecting a nucleated cell of some sort. Then viral proteins are going to be cut up by cellular proteases, namely the proteasome. Those chewed up proteins are going to transport into the endoplasmic reticulum where MHC is docked. And then those peptides get loaded onto the MHC peptide binding groove and then transported to the cell membrane. So MHC is like the protein logging system? Yeah, I think so. I think that's not a bad way to look at it. Like if you're thinking about an assembly line and, every 100 or so items gets like checked for quality, the MHC protein is kind of like that procedure. It's just a quality control mechanism by which a cell can display to the rest of the body, hey, I'm chill, nothing's going on with me, or I'm under attack, I'm being infected, something's bad is happening, or I'm turning into cancer, So it's like a way to be transparent with the rest of the body. Yeah. I've had some describe it as a window, though it doesn't actually make a hole in the membrane. I think that's why I don't like window as, because much like apoptosis implies that the cell explodes or pops, where it doesn't actually pops, it kind of dissolves into little blebs. I think there are some analogies that are not as good as others, and I think instead of window, I do prefer just like a signal. I know it's not a very romantic metaphor or illustration, but I think window is a little much. Now, how does it break? Ooh, we will get into that. Not too long from now, we're gonna get into that. Okay, so why do I love this protein so much, right? So it is immune meta defining. Basically, every virus gets gatekept by this, which is something that even interferon might not be able to do, right? So I'm working on a video behind the scenes, and I was actually surprised to find that some viruses don't actually care all that much about interferon. So interferon is an antiviral response. It's a part of the innate immune system, where you have a signal released by cells that are under attack that warns nearby cells in the immune system that an attack is happening. It's very general, it's very broad, it's not specific to any certain bacteria or virus, but a burst of interferon really kicks the immune system into action. And it surprised me that some viruses don't really care that much about interferon, but I think almost every virus I've learned about really cares about this protein because it is that centralizing of a immune force, I should say. So most of my channel is about the germ wars. This one protein is central to the germ wars. If you're a vertebrate, if you're, I don't know, a lamprey, I guess you don't care. But if you are a vertebrate, you certainly do care. And what I really like, again, this protein is so weird. It has a double life in the nervous system, and its double life in the nervous system has nothing to do with immunology in the brain. It's like not, because again, we talked repeatedly on this channel how the brain is kind of sort of differently immune privileged. Like you don't really see a lot of brain infection. You don't really see a lot of brain inflammation if you're healthy. And so for a long time, people thought that there was just no immune activity in the brain at all. When in reality, yeah, the brain's making quite a bit of immune protein, not in absolute volume, but they do express immune proteins in a regulated pattern. So it's not just randomly popping up, but like you'll have regions of the hippocampus that really like making certain immune proteins. And we're learning more and more that the immune system is actively playing a role in how your brain is wired. And so this protein that is so central to the germ story has this weird niche interaction in the brain where it's helping things like regulate how strong neurons fire, which I love. Like, so that's so weird. That's so weird, having been taught your entire like medical education, biology education, that MHC class 1s are the antiviral protein. They are the centralizing anti-infection meta. And then you later learn in graduate school this weird, weird other function that we don't really fully understand, but it's important for the brain in a brain fashion and not in an immune fashion. And that's so cool. I think that's really cool. Lampreys are in chordatus. Sorry. Not vertebrates. Okay. Jawed vertebrates versus jawless vertebrates. That's what I meant to say with lampreys. Sorry, my bad. Cancel me. I don't know animal biology. Sorry. Lampreys are jawless vertebrates. They don't have adaptive immunity the same way that we have adaptive immunity. Sorry, it's not vertebrate versus non-vertebrate. It's jawed vertebrates versus jawless vertebrates. This is why I did marine bio trivia with Octopus Lady on my 24-hour charity stream. Like, I don't know anything about animals. Dang it. Yeah. I got Uno reverse canceled. No, I was wrong. I was okay. I'll take it, dude. I'll take it. I was wrong about the vertebrate non-vertebrate split. It's jaws versus not jaws. Okay, so MHC class 1, as we just talked about, has a rich and complicated evolutionary history, right? Like there's some line between weird fish that don't have jaws and weird fish that do have jaws, where all of a sudden you get a lot of adaptive immunity happening. And I'm going to make a full, it's probably going to be like an hour long video about the history, the evolutionary history of the immune system at some point. I'm not ready to do it quite yet. Maybe it's a project for next year, but oh man, the history, the development of immunity is just insane. It's an insane story. And the thing about MHC is that it makes you mostly a unique and special snowflake. Like it is, the way that you inherit this is highly individual, and there's some science, although I cannot vouch to know how strong the science is. I think someone else in chat had alluded to the study that this is one of the, we'll say, not pheromones, but like there's some again, don't know how strong it is, but there's some evidence to show that I think the study was something like they had female volunteers sniff out the used shirts of male volunteers and found that the female volunteers preferred the smell of the used shirts of men that had a complementary haplotype of MHCs, meaning that there's some potential subconscious level of sexual selection that happens at the level of the immune system. And part of that might be, again, I would be highly cautious with interpreting any of this as true or not, but some of that might be due to the fact that we are trying to select for healthy partners who will have healthy children, complementary immune system stuff. Again, I cannot vouch for that because that's outside of my field, but it's just a fun fact that I picked up at some point. Yeah. Okay. So MHC class 1 is unique at so many different levels, right? I love weirdo proteins. There are, so what do I mean by weirdo versus beautiful? I want to make that clear. I think of ATP synthase, Clockwork did a video on it. Oh, one of the most beautiful proteins to ever exist, right? This turbine that is powered by an electrochemical gradient that, actually, is it right to say electrochemical? I don't really know. Whatever, it's powered by a proton pump. This wonderful turbine that is so central to all of human and general biology. It is highly conserved and central to the fact that we can generate energy from the molecules that surround us, right? Like it is such a beautiful protein, but I prefer MHC, not because it is more beautiful, but because it's just so weird. And we're going to talk about how weird it is. Right, so most animals have their own versions of this protein/gene family. And so what does that mean? In mice, we call the MHC class 1s H2K and H2D. Then those are two members of the MHC class 1 family. And in humans, we have 3. So already there's like this weird difference between rodents, who are kind of our cousins, right? Like primates and rodents are not super far away on the evolutionary tree. And yet we're already pretty different, right? They have H2K, H2D. We call ours HLA-A, HLA-B, and HLA-C. So we have an entire additional family member that mice do not have. And so this makes it really problematic for a consistent taxonomy. But I do think that in this case, we each have our own unique name for this protein because they're so different that you kind of need a different name. Otherwise, you're going to get lost in the sauce, right? Whereas a protein like ATP synthase, you just say human ATP synthase or mouse ATP synthase or, yeast ATP synthase. It's still the same protein, but these proteins in this family are so weird and quirky and unique, and they evolve very, very quickly that it does make sense that even though they do share a function, they actually are pretty funny and pretty different. Hey, Mylan, welcome. Yeah. So this presents a bit of a problem when relating the mouse and human immune systems, right? Like there's this conversation that is happening in academic circles as well as in the public about how well animal models can really capture human biology. And me, I am supportive of animal experiments where it makes sense. And you really have to make sure that when you do pick an animal model, if you do choose to use animals in research, that you are doing so responsibly. And part of that responsibility is making sure that you are not overgeneralizing your results, right? So, if you do MHC research in mice, you have to always acknowledge that humans might be a little funny and a little different, right? There's already a big difference. We have one additional, we have one additional protein family member than mice do. That doesn't mean you can't do mice research and then have it relate to human. You know, like you can, you just have to be a lot more careful with what you can and can't say. So there's my animal spiel there. Is there one extra in humans, or all three totally unique and unrelated to the rodent equivalents? They're similar, but I think we're going to see in the evolution of primate MHCs, like not even talking about rodents, but we're going to see how crazy different, you know, even within our monkey cousins, like it's already going to be kind of funny and a little different. So it's not that they're so like unrecognizably different. If you look at their crystal structures, there's a good amount of similarity, but the context by which they evolve in is going to be you cannot assume that it is the same, is what I will say. I think that is fair. Yeah. Okay. So MHCs/HLAs in humans, these are the most polymorphic human genes. So you have three MHC class 1 genes, HLA-A, B, and C. Each of these are highly polymorphic, meaning there are thousands of different variations in the human population for each of these family members. Of all the human genes, this family has the highest amount of diversity. So between you and even your sibling, you might have different MHC genes, which could be radically different than somebody on the opposite side of the planet, right? That is in contrast with, say, the ATP synthase gene, which is not that diverse, right? Very little diversity there. So ATP synthase has extreme evolutionary pressure to stay the same, as its function is very important, right? Nature is not going to be tinkering with energy generation. It doesn't need to, right? If it's working pretty well, you know, been refined over pretty much all of the history of life, there's not really a need to do too much experimenting. It works really well as is. However, MHC's job is to fight evolving pathogens, so it makes sense that it is just as varied as viruses are going to be, right? So this is what we call an evolutionary arms race, where pathogens prey on us and we evolve and adapt to combat them. And a lot of the genetic diversity happens right here in the antigen binding groove, right? In the place that MHC binds to those peptides. Because if we as a human species, if we all had the same MHCs, we displayed only the same set of peptides, we're going to be vulnerable to attacks by viruses who might not make those particular peptides, might hide those particular peptides, et cetera, et cetera, et cetera. So that is the polymorphic, the liquid, the complicated nature of MHC. Okay, so where is the peptide groove? I'm showing it to you right here. I've got arrows here. So this little region, this cleft, this pocket, this is where a lot of those sequence diversity is going to be located in. Of course, there's diversity elsewhere in the protein as well. Oh, sorry. Oh, yeah, okay. Yeah, right. My mouse should be on screen. So here is the pocket, and you're going to see between individuals, between species, that the sequences of amino acids that outline this particular pocket are going to differ a lot between people, so that every little, you know, every person has their own special unique immune fingerprint, so that we can all be differently good at expressing or displaying different peptides, right? So yeah, this little portion of the protein controls which peptides are presented by each MHC. So I might be really good at displaying glycine, alanine, leucine, tyrosine, phenylalanine, tryptophan, and you might be really good at displaying serine, tyrosine, valine, proline, glycine, right? I'm just making peptides up, but that's kind of how you should imagine this unique immune fingerprint. Yeah. So some versions of the protein are going to be slightly better and worse at displaying certain peptides. But this means that even though on an individual level, right, like we don't have the capacity to display every single peptide in existence, right? If we're thinking about all the potential peptides that can be generated between 8 and 11 amino acids long, that's going to be a lot more different combinations than our proteins can handle. And it's not one-to-one, by the way. Like this groove, for each MHC family member, this groove can bind hundreds to thousands of different peptides. It's just which peptides that it prefers to display are going to be a little different between individuals. Okay, so at a population level, this means that we are much more likely to survive due to having a high diversity of immunity pre-built into our biology. So why do I say pre-built into our biology? Anybody can tell me kind of why I highlight that this is a genetic level immune defense? What's a immune level defense that is not hard coded into our genetic code? I joined midstream, so I don't have a lot of context, so my questions might be kind of stupid. There's no stupid question. You're in a graduate to undergraduate level lecture about a very niche interaction with one specific protein. Any question that you have here is going to be pretty smart. I can guarantee you that. Antibodies and T-cell receptors. Antibodies, yep. Adaptability, TCRs, yep. So I've said it multiple times on this channel and in multiple videos, but something like T-cells and antibodies, they are different than this hard-coded immunity, right? They adapt to new threats because they can scramble their DNA. So they are complementary in their approach. So I wanted to illustrate exactly how crazy the diversity of HLAs can be. So we're talking just humans now. So HLA-A, 9,000 different inheritable versions. HLA-B, 11,000 versions. HLA-C, also 9,000 versions. These are very recent. I just looked these up in the HLA database. I think even like 5 years ago, it wasn't that many. I think like HLA-C 5 years ago was like 7,000. So every day more of these sequences are being found in the human genome. I forgot, how does a high polymorphism help on the population level? So it gives us a pre-built immunity, a bias towards being able to counter certain viruses or certain bacteria slightly better than other people. So you can imagine if we had all the same immune system, that a virus just needs to figure out how to defeat one person and then it gets free reign to everybody else. This makes it so that a virus is going to encounter a very unique individual every time it tries to infect somebody. Yeah, so it's pretty crazy. So not only are there thousands of versions, remember you get one copy from mom and one copy from dad, right? You have one copy from mom and one copy from dad. So when you think about the combinations, right? So there are 3 family members and you get 2 copies, one from mom and one from dad, each who can have a different assortment of these genes, and you get a truly staggering amount of diversity, hypothetically speaking. There are also HLA-Ds, E's, and F's. These are non-classicals. These are not the classical class 1 MHCs. There are hundreds of versions of those. They're not really doing the antigen presentation stuff, so we're going to ignore them, but I just wanted to point them out because they are included in the HLAs. How does this affect autoimmunity? What about organ transplants? So I'm gonna be so real right now in terms of like organ rejection and stuff. I don't have a background in that kind of stuff, so, and I don't want to like get very basic things wrong. So I'm just gonna say I know these are involved in organ rejection. I don't remember exactly how. It's a good question though. So let's do some math. How many potential combinations, unique human immune fingerprints could there be mathematically? And this is a napkin, back of the hand napkin level approach. So you can check my math. This is my calculation because I couldn't really find an answer online for some reason. So if you can get one copy from mom and dad, that means there are 9,000 squared possible different arrangements. I think that's correct. Is this correct? Because whether or not you get it from mom and dad might not matter. So it might be less than this, right? Because I think this is assuming that order matters when it doesn't. Somebody who's good at math, please help. I just realized that order doesn't matter, so this number is less, but not that much less, I think. Might need to divide by 2. Yeah, that might fix it. That might fix it. So 9,000 squared, 11,000 squared, 9,000 squared, and then when you multiply these, I believe you get 10 to the 23, and then you divide by 2, which is still going to be about 10 to the 23 or 10 to the 22 possible theoretical combinations. The reality of genetics makes this not true, right? But I'm just saying, like, mathematically speaking, if you assume ideal separation, if you assume independent assortment, which is something that you can't assume here, yeah, basically, I'm just saying that for the genetics heads out there that like will be like, this is not actually true because of genetic linkage. Yeah, you're right. But I just, for the point of like expressing how potentially diverse this makes your immune fingerprint, 10 to the 23 is like a huge number that I can't even conceive of, right? Now, the realities of genetics really constrains this number to a much more reasonable level of freedom. I don't know what that number is, but it's a lot less than 10 to the 23. But it just, I'm trying to illustrate the idea of complexity here. Genetic linkage? Yeah, so the idea of genetic linkage is that some genes are inherited with each other because they are near each other on the chromosome, right? So if HLA-A and HLA-B are right on top of each other, then you're not going to get what's called independent assortment, which is an assumption that your genes, separate individually during meiosis. It's a bit of a complicated subject, so I'm not going to get too into it, but just know that the closer genes are to each other, the more likely it is that they are inherited with each other. So there are certain versions of HLA that happen to be co-varied with other members of the other family, so it's not quite this free, but that's the idea. That's the idea here. Okay. So the fact that this one gene has so much genetic diversity is a living reminder of the long history of the germ wars. And I just think that's rad as heck, right? This diversity evolved out of a need to constantly be on top of the anti-pathogen war, right? We need, or rather, the fact that you have this little immune fingerprint means that you are living proof of an ever evolving back and forth battle between viruses, germs, fungi, worms, what have you. And I just think that's really cool. I just really think that's really cool. Anyone not hearing anything? Is my mic cut out? No, from OBS it seems fine by me. Okay. So I can hear how rad it is. Thank you so much, Surprise Pants. Okay, so I wanted to contrast the immunology of MHC and T cell receptors because I don't really talk about MHC all too much on the channel. So, MHC, these are on all nucleated cells. They display internal antigens to T cells. The diversity is hard-coded into your DNA and is inherited from your parents. The diversity determines the efficacy of displaying certain antigens over others. And this provides us with a population level protection, right? So you heard of herd immunity. It's kind of a similar idea where diversity is our strength in this case. We don't want the same immune system because, again, it just takes one lucky virus. And that's, an example of this is like the banana situation. Oh, the banana situation is crazy, right? The fact that all of our modern culinary bananas, the ones that we eat, are clones of the same stock means that it just takes like one virus or one fungus that can get a head up on our pesticides, our treatments, and the biology of the banana to just ruin the entire banana ecosystem. So there's a lot of biological engineering that's interested in figuring out how to preempt the collapse of the banana industry, right? So just like the banana, we would benefit from having this diverse immune system at a population level. Yeah. R.I.P. Gross Michelle, the banana we can't have anymore. It's so true, so true. Although I think maybe Hank Green got the chance to have one, which I thought was very cool. I think that's pretty cool. All right. So how does that compare with the T cell receptor? So the T cell receptor, that's only on T cells, duh. It binds to MHC. If antigen in the binding pocket is foreign, it triggers an immune response. You're going to have degranulate. Is that correct? Is that the right word? I don't remember for the T cell. In any case, the T cell is going to release a whole bunch of chemical messengers that are going to force the cell to do some apoptosis, right? So this is cell killing by the immune system. The diversity here is generated by the T cell level by VDR recombination, VDJ. Oh my God, the typos today. By VDJ recombination, which is something that I've covered before, but essentially your T cells will scramble its own DNA to generate new proteins to be better able to detect when a cell is infected, right? So that diversity is generated and not inherited. And this provides you with individual level protection. So that's your individual immune system doing individual immune system stuff. Yeah. All right. So I just think this is really cool because this is kind of clamping down. This is like, to me, this is like a top and bottom jaw, right? The bottom jaw MHC receptors, pre-built, pre-built hard-coded immunity. And then the T cell receptor, which is procedurally generated, Minecraft world kind of like, based on a set, on a randomness. And I think having both of those jaws clamp down onto pathogens is a really effective strategy, as is evidenced by the fact that we are still alive today, right? This system works pretty dang well. The recombination is coming from inside the PowerPoint, that's true. It's recombining my spelling errors. Okay. Oh yeah, I do talk about vertebrates and stuff. So the evolution of this protein, my favorite protein, goes hand in hand with our history as vertebrates, right? So jawless fish like lampreys, these guys opted out of the whole modern adaptive immune system thing, although again, they do have their own weird, quirky version of adaptive immunity, which I hope to cover at some point. They don't have MHC, they don't have T cells, they have, a really weird version of this that I don't fully know about right now. But then at some point, between jawless fish and jawed fish picked up two very important transposons called RAG1 and RAG2. These are jumping genes. They pop in and out of your genetic code, and it is thought that RAG1 and RAG2 eventually became VDR recombination again. What? I don't know why I thought VDR here. In any case, the system that gives your T cells the ability to generate near infinite complexity comes from this RAG1, RAG2 transposon that we inherited back when we were like crocodiles, essentially. Probably a little bit before crocodiles, although, again, I'm not an animal person, so I don't fully know. Jumping genes, yeah. There are some genes that you have that can just leave your chromosome and pop back in. The process is highly regulated, but it is interesting that the genome itself has a kind of instability at a molecular level. And I think that's pretty neat. So from this, we have MHC's base body plan established from like 500 million years ago. So even though I talked a big game about MHC being so diverse across species, the basic body structure of this protein was established a good chunk of time ago, right? 500 million years ago. So from there, genetic diversity with regards to immunity radiated out like no other gene, right? Immune genes are very, very diverse across species because of how pathogens challenge us at a survival level. All right. If jawed fish have them, it would have been way earlier than reptiles. Yeah, I included an alligator or a crocodile, don't know which one that is, purely because I could show that image without copyright. Because I was like, what picture of fish do I show? And then a lot of them came with commercial restrictions, so I didn't want to do that. Okay, so MHC was so centralizing of a meta that viruses evolved multiple ways to sabotage it. And here I think we're going to have a lot of fun with this exercise. Okay, chat. So I have here a summary, the same summary I showed earlier about the antigen presentation pathway. I summarized the entire process by which this sentinel protein gets made, loaded up with a peptide, and shuttled out to display the infection status of a cell. I want you to be the virus, right? Get inside the head of a virus. How would you sabotage this pathway? And I want to see, chat, if you can be as clever as viruses can. So I think on the next slide, I have quite a few ways viruses have historically sabotaged this meta. And I want you to just try and capture as many of the strategies as you can. And I'd be really impressed if you were actually as clever as viruses. So go right ahead. And I'm going to take a moment to hydrate while you sit on that. Go to the fish market and photo yourself? Nah, that requires touching grass. When I could be making content. I hate boneless animals like Brazilian electric eels. We wouldn't never have known they even existed because nothing would fossilize, even if we wouldn't know they were electric. Yeah, I could not imagine studying animals. Sorry, reptiles, amphibians, and reptiles. Oh, sorry, I thought that was a typo. I don't know anything about animals. I am the worst biologist to the average person because I know nothing about animals. Mimic host proteins. Say more about that. How would mimicking host proteins sabotage this pathway? Denature the proteases. Oh, interesting. Find peptides that bind to MHC and disable them. Okay, interesting. Shove other proteins in to clog it. Oh, very cool. Very interesting. Stop proteolysis. Okay, so stop the chewing up of protein. Mess up protease synthesis or structure. Also an interesting idea. Sabotage the proteins that load up MHC. Maybe like herpes, blocking transporters. Okay, somebody has a little bit of a spoiler. Someone has been hitting the books. I forgot to remind people, but this is like an exercise for people who don't have biology background, because if you do, then you know the answers already. Okay. Bung up the endoplasmic reticulum. Yeah. Okay, you guys are generating a lot of interesting ideas. Don't express MHC in the first place. Yeah, keep the MHC bound to one protein. Interesting. Maybe integrating MHC or parts of it in my own genome. What do you mean by that? Integrating MHC. Not to say you're wrong. I'm a little unclear as to what the language means. Yeah, you guys have come up with a lot of good responses, and maybe you have come pretty close to capturing everything on the next slide. So I am going to move on here. We're theory crafting new viruses. I think every way where here's the thing. I think probably, this is not an exclusive, an all-inclusive list, what I'm about to show, but I think over the millions of years that this battle has been raging, I think viruses have probably figured out most of the ways to actually do this. So let's have a look. And again, this is non-exhaustive, so if you suggested one that wasn't on this list, that doesn't mean that you're wrong. It also doesn't mean that a virus didn't already do it. This is just everything that I could find in the short amount of time that I had to prepare this presentation. Okay, non-exhaustive list. Number 1, pull MHC out of the membrane. So make a protein that yanks MHC straight off of the membrane, hide it from the immune system. 2, stop MHC from getting to the membrane. So everybody who said something about the endoplasmic reticulum or the Golgi, you get a point there. So jamming their ability to make it to the membrane. You could destroy MHC, the protein, right? So Marlon mentioned earlier ubiquitin. So ubiquitin is a little tag that enzymes put on proteins that are destined to be recycled. So you could make a protein that recruits host ubiquitinase, enzymes that add ubiquitin onto things, and get it to tag MHC to destroy it. That's also a possibility. I saw somebody in chat recommend turning off MHC production, so you just stop the cell from making it at the genetic level, right? So stop the mRNA from being made into protein. You can block TAP, which I believe was spoiled in the comment section. So TAP is the antigen transporter that moves antigen into the endoplasmic reticulum. So there are classes of viruses that stop this from happening. You jam up that channel, and you can no longer stick those proteins onto MHC, so they become useless. Right, you can block the proteasome, which multiple people have suggested. So the complex that chews up the protein, you can go ahead and jam that. Viruses do that all the time. Yeah, no harm, Vietran. I'm not roasting you. I'm roasting myself for not reminding myself to preface that these, you be the scientist, you be the, whatever. These are for like the general public. But I do appreciate the enthusiasm. I do appreciate the enthusiasm. What else we got? So block the peptide chaperones. So there are proteins that are inside of the endoplasmic reticulum that help MHC fold in a way that allows them to load in that peptide, and there are viruses that jam the chaperones. You can block beta-2M. So I didn't talk about this protein very much at all, but let's see. Beta-2M, that's this That's this protein over here, beta-2 microglobulin. This is a protein compound that stabilizes MHC expression at the cell surface. So if you remove this from MHC, this protein is then going to be destined to be re-internalized into the cell. So this is kind of the protein anchor. So I hope to illustrate here that the germ wars are brutal and bloody. Just like look at how different these approaches are to sabotaging MHC, right? You've got a long genetic history of this battle. And there is one anti-meta strategy that I want to point out that people are spoiling in chat. That people are spoiling in chat. And that is the fact that there is a class of cells that was introduced in evolutionary time to counter this counter, right? So if all these viruses are making this anti-MHC strategies, pulling it out of the membrane, decreasing MHC, removing its anchor, stopping it from getting to the membrane, pulling it out of the membrane, stopping it from being made at all, destroying it, etc., etc. All of these things converge on one idea: the removal of MHC from the cell membrane, right? The cell is no longer making MHC. And so there's a special type of immune cell that was evolved to counter this meta. So this is a counter meta, the natural killer cells, okay? Natural killer cells. So these were cells that were pressured into evolving to counter the counter meta, right? This is called the missing self hypothesis. NK cells can actually detect if cells are missing MHC, which is another sign they could be infected, right? So I'm not going to go through the missing self hypothesis, but basically there's a logic gate, right? The NK cell binds the cells, and if they don't sense that the cell is making MHC, they blow that cell up, just like, or depending on the natural killer, they might blow the cell up, they might alert the immune system. They're going to counter this counter, right? How would you counter this counter? Okay, ignore this, ignore what people have said in chat. Okay, this question's for the general public here. If you have a cell that is evolved to counter the counter, a cell that detects a lack of MHC, you be the virus. Okay, you be the virus here. What would you do in order to counter this counter meta? What would you do to counter this counter meta? I want to know, what would you do to counter the counter meta? Don't look at because people have spoiled it, so don't look at that. Don't look at that. I just want to say this content is just perfect to procrastinate my research without feeling guilty because I'm at least learning something. That's awesome. I love that for you. I love that for you. Yeah, a whole lot of countering going on. Exactly. That's why I love immunology. It's it's essentially like watching a fighting game or Dota 2 or any competitive fighting game, any competitive game at all. It's really, really interesting, the counters that the immune system has developed. Yeah. So what do people think? Make my own MHC that presents whatever I want. We're talking about from the viral perspective. So the virus is countering the natural killer cell somehow. Make something that presents an MHC-like antigen? So find a way to make some MHC. Produce a dummy protein to trick it. Ooh, use a shell. Produce it yourself. Oh, counter is currently at 3. Yeah. Don't attack the MHC production or display, and instead make a protease that stops your specific protein from being displayed. Ooh, that's really clever. I like that. Yeah. Okay, so you've had some time to get in the head of a virus. Let's see if you're right. So if NK cells are detecting a lack of MHC, give them what they want, an MHC biomimic. A fake MHC that looks real but doesn't actually display viral peptide or trigger an immune response. A devious trick developed by the herpes viruses, okay? So this is part of the reason why you should be hyped for the eventual herpes virus video. Those viruses have insane immune evasion mechanisms, right? So here's my worry about covering herpes virus in a video, because I don't think a lot of people will be interested, but I want you, my streaming audience, to know that of the viruses that I personally have studied, herpes viruses have some of the craziest, wildest counters I've ever seen, right? So when eventually that video comes out, remember this stream and watch that video. So I have an additional treat for you. Let's take a poll. Which of these is the viral MHC and which is the real MHC? I'm doing a poll, is the real MHC. See if you can tell. And I'll do left and right. If you were an immune cell, would you be able to distinguish between the two? Which looks real? Which looks real to you, huh? Which is the viral mimic? If you were a natural killer cell and you were trolling the immune system, would you be able to tell? They look pretty similar to me. Right? All right. Which is the real MHC? That's what this poll is. And this poll is awesome. Look at this poll. You can't tell it's 50-50. You're essentially going off of a vibe, right? It's luck, right? So I want to point what we're looking at here so that we're understanding how this is happening, right? So here I have outlined the antigen binding groove. So I'm going to go back a few slides. So this groove up here, we're going to be looking at this. That picture is this from an aerial view, right? So this little pocket, we're going to look at this pocket from the top. That's the top, right? This is where the antigen sits. And you can see, look at this antigen binding groove. Like, look at this particular shape. These two little curls followed by this sheet. It is, come on, that's identical, man. That's identical. And then I drew in a little sample peptide in the middle. Isn't that incredible? That's incredible. Right? That's incredible. Okay, let's just appreciate that. The part of the receptor that displays antigen, it looks identical to the real one. So which is the real one? I didn't even reveal it. I didn't do it in the slide. This is the real one. The answer is right. The right one is the correct one. The left one is the viral mimic. Isn't that crazy? That's crazy, dude. Average NK cell answer. That's crazy, right? It's similar enough where, I mean, not that you would have the knowledge, right? But there's no difference between these two proteins. There is on the cytosolic side, right? You can see that this cytosolic side, or the bottom of the protein, which we're seeing it here, versus the bottom of the protein, which is here. Like, these are different, but the part that is doing the MHC function is like identical between viral and cellular, right? When they say biomimic, it's incredibly uncanny. I mean, look at the detail. Look at the detail. Surely it wouldn't have the exact amino acid sequence, even though it has the same structure. I actually don't know how similar the binding pockets are. I wouldn't be able to tell you. But I mean, like, even down to this detail, like, look at how this helix goes into the screen, right? It points downward into the screen. This helix also points downward into the screen. It's a slightly different angle because the structures are slightly different. But I mean, if I were an NK cell, I would not be able to tell, right? My receptor glides along this groove. It probably says everything is okay. This is how crazy immunology meta is centered around MHC, that you have counters, and like, God, it's the layers, dude, the counterplay, the nuance, man, the meta is just so interesting. I love it. It's so good. So do natural killer cells only look at the groove? It's more than the groove, but I think from the perspective of the receptor, the groove is like the most important bit. So cool. So sick. So what's counter to this? I mean, the rest of the immune system, right? So even though herpes viruses can counter here, you know, we have B cells, we have antibodies, although herpes, again, the herpes viruses are some of the most immunoevasive viruses I've ever seen in my life, because once you have them, they're with you for quite some time, you know? And they can only really do that if they're really good at countering, right? So we do have other antiviral tools, but herpes is like really good at fighting our antiviral tools. What does MHC stand for again? It stands for Major Histocompatibility Complex. Do you think they learned through evolution or by somehow acquiring the sequence from the host? This is an excellent question, and I don't know. I hope to know the answer by the time I make the herpes virus videos, because, you know, horizontal gene transfer does happen between host and virus. So I'd be really curious to know whether or not this is an example of horizontal gene transfer. Yeah, I don't know that answer. It's a good question. It's a good question. The Virus's New Groove, true. Good movie. Okay, so I hope I have successfully demonstrated why this is my favorite protein, right? It's because the antiviral meta is built around this protein. And again, you know, I'm not as familiar with interferon and the vast variety of ways that viruses attack interferon signaling, but I did recently learn that there are some viruses that don't really care about interferon, whereas basically and I could change my opinion on this later, but basically every virus that I have heard about has some counter to this because it is so centralizing. That's not to say this is more important than interferon, it's just that the evolutionary pressure to evade this is very, very high. So viruses can't ignore this if they want to stay in the body for any amount of time, right? Like you get those T cells rolling in and the viral infection is going to be seriously challenged. So MHC is also the reason why I have a channel built around immunology today, right? Like, I remember my advisor told me this story in a talk. This was before I was even a student of my advisor, right? Just in a faculty talk, we were all trying to see where we wanted to rotate, and my advisor told this beautiful story about this protein, and I added on to all of this that this protein is doing something completely different in the brain. I was like, you know, I'm in. I am pot committed. I want to know all about the weird overlap between immunology and the brain, because this is a very weird protein and it's doing something very weird in a very weird part of the body, right? The brain is very, very weird. It's got its own set of unique exceptions, counterintuitive rules, etc., etc. And so, yeah, it's just, this is a very important protein for my history as a student of biology. And I hope that me communicating this has evoked in you some of my excitement when I heard about this protein so many years ago. Yeah. So can interferon be used to treat disease? I believe it is used in some circumstances as a medicine, but I'm not too sure because I'm not a doctor, right? I have heard of interferon being administered to activate the immune system. Does it do completely separate stuff in the brain? It absolutely does. It absolutely does, because there's not a lot of viral infection in the brain, right? I mean, if you have a viral infection in the brain, you're in for a rough time. The brain still makes it. It makes quite a few immune proteins that interface with how well your neurons talk to each other. So, for example, the complement proteins that are related to MHC, the complement system is like an anti-pathogen defense that's in your blood. So it's a very fast-acting way to counter pathogens that are in your blood. Those same complement proteins that are a part of this immune response are doing things in the brain like trimming your synapses. So when you are born as a mere babu, each of your neurons is incredibly connected to its neighbors. Your neurons actually have too many connections. And then as you grow older, as you mature, as you age, your microglia, the macrophages of the brain, will come around and chew up the extra connections that you don't need, the inefficiencies between, it's not really fair to call them inefficiencies, I shouldn't call them that, but like the connections that you don't need. And how do microglia know which synapses to chew up? They find immune proteins like the complement proteins. The microglia detects those complement proteins that are expressed on extra synapses that don't need to be there, and then that's how they know to chew those synapses up. Like the intersection between immune stuff and nervous stuff, oh, I could talk all day and night about it. So cool. So much fun. What does histocompatibility mean? Does it have anything to do with histones? I believe it has something to do with organ compatibility, but I'm not super sure. Wait, if everyone has a different MHC, how does the virus mimic every person's? Does it detect for a non-variable area of the protein? That is also an interesting question. I think there's some wiggle room. I'm guessing here. This is my best stab at answering your question, because it is a really good question. There's a lot of variability in that antigen pocket, but at the same time, it still has to function as an antigen pocket. And I don't know whether NK cells are discerning enough to have evolved lock and key level recognition of that receptor, right? So everything is a little bit of putty. Everything's a little bit flexible, is what I would guess. The brain is very weird when it comes to the immune system. Yeah, due to blood-brain barrier. Yeah, and we recently learned that there's a secret backdoor into the brain through the, you can sneak immune cells into the brain without having to go through the blood-brain barrier through specialized channels that we're learning a lot more about. So even that relationship is being challenged. It's such a cool, interesting, rapidly evolving area of research. It's good. It's good. This protein's so cool. I'm glad you found this info, and because of that, now we have this amazing channel. Thank you. You're really inspiring me as another biology student that doesn't like animals. Yeah. I just think there's so much to learn about animals, whereas like the things that you learn about one animal might not be applicable to another animal. But why I like cell and molecular biology is even though yeast and humans are different, there's a good amount of overlap at a cellular level between yeast and humans where it doesn't feel like you have to learn a new thing every time. That's my perspective. My favorite professor, anatomy, actually studied MHC and was a neuroscientist. That's awesome. I might know them. They almost certainly don't know me, but I might know them if you wanna drop their name. Yeah, I'm so curious as to who you know, because there's a chance, because this is like, neuroimmunology was like my focus for a while, that I might know who you're talking about. Yeah. Can an overproduction of this cause an issue in the brain, like dementia or something? It's an interesting question. We don't know a whole lot about how this protein functions in the brain. As with all things in biology, you really want the right amount, which is a cop-out answer. Too much of anything is bad, too little of anything is bad. That's the general rule of homeostasis, right? You want the correct amount for whatever you're talking about. What if the synapses are connected to a neuron that does something important? So active synapses that are being used, are not making the complement protein. Yeah. Does it destroy the synapse or the entire neuron? Microglia just eat the synapses, just the extra connections. Excuse me. Yeah. Are you going to make a video on MHC in the brain? At some point. I think I would love to do a weird intersection of neurology, neuroscience, and biology in the brain. It's just, I'm still playing with how to do it. Like this stream is like practice because the ideas are pretty complex. I think anytime I cover neuroscience on my streams, I tend to get fewer viewers because the amount of background you need is a lot higher. Because like molecules and proteins and cells, it's like you can see them, right? Neuroscience is abstract in a kind of way where it's like neuron firing rate and connectivity and networks and feedback loops, and those ideas are a lot more challenging to make palatable. Whereas like part of why I love cell and molecular biology is I can just go, hey, look at this protein, and then you can see it, right? So I'm still playing around. I'll do a couple journal clubs around the idea, and then when I've had enough practice on how to communicate this, I will probably make a video at some point. Yeah. Wait, sneak through the BBB how? Could you mention the protein or thing involved? I forget the actual thing. It's in my Discord. You can do ! Discord if you're not a member already. I posted a link of a paper that discovered a new route, a little pathway that immune cells can squeeze through to get access to the brain without the blood-brain barrier necessarily having to be weakened. Yeah. Yeah. Love this stuff. Love this stuff. All right. So, which channel, the backlog? I think it's in stream discussion. You might have to scroll up a bit. All right, so let's move on. Let's do a bit more yapping on this protein, why don't we? Okay, so let's talk about the paper for today. The primate major histocompatibility complex as a case study of gene family evolution. So if you have been paying attention to my videos, I've been on a real gene birth, gene death evolution kick recently. It's kind of been the theme of 2026, as I've been thinking a lot about genes, which is something that I didn't think I would do because I was not a good genetic student, but you know, it's just where I think it started from the E. coli evolution video where I first started to get interested in gene birth, and then gene birth became its own video, and then gene birth led into abiogenesis because those ideas are also kind of related. So I've been really reading a lot more about how genes are born and how they die and how they relate to each other. So that's my current scientific obsession, I think. The problem, though, is that I'm a really bad evolutionary biologist, so I picked this paper because I thought it was a good way for me to challenge myself, but then I read the paper and I was like, I so regret picking this paper. Not that I regret making this journal club, but I'm like, man, I am like not cut out for this. So what you're going to be seeing is my best attempt at interfacing with a very complicated topic. So much like I recommend students, in order to deeply learn a subject, you should try your best to teach somebody about it. So this is me brushing my teeth and interfacing with a part of biology I'm not as familiar with, trying to teach it so that I myself can learn it. Okay. And I wanted to do this because I know from my history as a cell and molecular biologist that this is a weird, funny protein. It does all sorts of crazy things. The immunology is like crazy, but I don't really know how the broader connections of this protein reach out into evolutionary time. So I wanted to get a sense for how we got to here and get a sense for what MHC was like in the past. And we'll see if I am able to answer that question as we get into this paper. Okay. I'll head out now. Thank you for the lovely presentation on MHC. Thank you for dropping in. I've always loved your stream format. Thank you. It's like an interactive conversational presentation. Yeah. I call it journal club. In reality, real actual journal clubs are not going to be this interactive because people have experiments to do. But that's why I love doing this for YouTube, because we can have these conversations. And also, this is my anti-content rant. You're giving me the soapbox, so I'm going to take the permission to step on it. I want to provide people with a venue by which they can engage with content in a deep and meaningful way, because I am selfish and I demand your attention. I think it's good to pay attention in a time, my attention span is also brain rotted. I'm also scrolling on YouTube shorts, right? But I think the more content that people reward that is engaging, I think the healthier it is for a content ecosystem. Let's just put it that way. Is it best if I watch part of the stream I missed, or can I join in now? You can join in, whatever you want, man. Like, the beginning, I think, has really interesting context and history of this protein, which I absolutely recommend that you watch. But if you want to just hear about, if I'm second monitor content for you right now, you can just leave me on and then watch the rest of the VOD later. Because it is like a lecture style, I think. I have a pretty good balance of talking with chat and actually getting on with the presentation. Okay. I was going to ask on Discord what exactly a journal club is like. I was playing with the thought of starting one at my community college. That's awesome. I love that. You would typically do a journal club with either people directly in your lab, but if you don't have a lab, then people who are in your department. So you would basically pick a research paper, and everybody. Here's how journal club was run when I was in journal club. In my lab, and again, it could be your department if you don't have a lab, every person was assigned to do journal club this week. So in a month in advance, I'm like, okay, this week I'm going to do journal club on Wednesday. We're going to gather at noon. We're going to order some lunch, and we're all going to watch a presentation on a particular paper that is interesting or important. And everybody's going to read the paper beforehand, although not everybody reads the paper before, but you should. One person makes a presentation that's kind of like this, although a lot more suited to researchers than the general public. So adjust your presentation style appropriately. And then you talk about the paper, you challenge the authors, you do the heavy lifting, and then your co-department people or your co-lab members will offer their feedback. And then you kind of figure out what the paper actually tells you, because that's kind of hard to do on your own. And it requires like your PI, your professor, other perspectives, because your PI is not necessarily the strongest person in the room, right? If you're covering a paper that is multidisciplinary, like a biochemistry paper, and you're in a biology lab, maybe there's a postdoc who has both backgrounds, and you kind of all get together, you share your expertise, and you determine what can I take away from the paper. A common trap with journal club is a lot of people view it as, how can I crap on this paper? How can I criticize this paper? How can I seem smart in this room of smart people? When the goal of journal club should be, what is the truth of this paper? What do I trust from this paper? So that's your journal club 101 idea here. Yeah. Okay. All right, let's keep going here. So a fast evolving gene like MHC is going to have some very weird consequences, right? We just talked about the fact that there are, on a napkin math level, 10 to the 23 unique ways for 3 MHC genes to be inherited. There's very little sequence conservation by design, right? So what does this mean? It means that between you and me, mathematically speaking, if we roll the dice and we make a bet, I'm betting that my 3 MHCs are going to be different than your 3 MHCs. Just gambling, I'm willing to take the bet. I'll bet $100 that we have different, that all 3 of our MHCs are probably going to be different, you and me chat. Now, if I had a huge streaming audience, then maybe I wouldn't take that bet because there is overlap between humans. But in a lecture hall, virtual lecture hall like this, I would be willing to bet that I'm a unique snowflake amongst you. But this, again, we talked about, this little sequence conservation is by design, right? We benefit from the fact that this is our immune fingerprint, that each of us makes this protein, but this protein is a little bit different in each of us so that we can have, on a population level, a robust defense against viruses. So I feel like this paper doesn't really have a real central story, which is also kind of fun because we can just watch this evolution happen as we discover what they did and like revel in the history of this protein. So unlike my other journal clubs, I was not able to pull a coherent story because this is so far out of my comfort zone, but we'll go ahead and see what we can learn from here. So yeah, MHC evolution across our close cousins is already really tough to study. So here we have a tree, right? An evolutionary tree of us on top, human, and the rest of our simian cousins, right? So we're in the grouping of apes, right? Our closest cousins are the bonobos and the chimpanzees, gorillas, orangutans, gibbons, and then we get into old world monkeys, so baboons, macaques, new world monkeys, marmosets, and then things that are technically primates but not simians, I think, like lemurs. So this is a tree of our relationship with other primates. And on the horizontal, we have a map of the different MHC class genes, I should say, right? So if we zoom in, we can see we have A here, right? This MHC HLA-A, I believe, because this is also kind of hard to understand for me. We have B and C here as well. And we can see that the major class 1s are pretty decently conserved, right? These are filled in, so they're fixed. So they're pretty stable in our genome. I believe that's what that means. And between us, chimpanzees and bonobos, like the MHC class 1s are pretty much the same, the Bs are pretty much the same, and the Cs are pretty much the same. These other guys, I believe, are class 2s and class 3s, and also non-classical versions of the genes. And you can see there's a lot of variation, right? So in certain species, like, let's see, where's a good example here? If we just take a look at MHCA, we can see that, well, what are we here? In gorillas, the HLA-A is unfixed, right? It's not fixed, so it might be a little unstable in gorillas. That's how I'm interpreting this, because this is pretty hard for me to understand. And then, yeah, there's a lot of differences. And just, even if you don't fully understand what this graph is saying, just take a note and just understand that this family of immune system proteins is really different across the different primate species. Why should sapiens be on top? Just so that we can easily compare all the other primates to us. There's no objective reason why, it's just for better communication. All right. So when we only look at MHC class 2s instead of class 1s, so yeah, so sorry, these are MHC class 1s, including the non-classical. So the classicals are A, B, and C, and then the rest of these, like F, V, P, G, these are non-classicals. These aren't really the antigen presenting ones, but they are related. So there's a lot of variability here. You can kind of get a feel for that. But when we look at the class 2s, we actually see that things are a lot more stable, right? If we look on this end, this right-hand side, we can see DMB, DMA, DOA. Like there's a lot of similarity on this axis, which is kind of interesting. I don't know what to make of that. I don't really know a lot about class 2s. What do the classes mean? So class 1s are MHCs whose job it is to display the internal antigens of a cell. So, you know, if a cell is infected, a class 1 protein, class 1 MHC protein will display that virus on the outside. A class 2 MHC is only on professional antigen presenters, so macrophages, microglia, dendritic cells, cells whose job it is to sample the environment, to eat proteins in their environment and display those. So they're similar proteins, it's just they both have slightly different functions. We aren't lacking anything on MHC2. It certainly looks that way. Certainly looks that way. And also, some of these gaps might not be like they're missing them. It might be that we haven't sequenced enough of their genomes to actually find them. So take the lack of certain genes with a grain of salt here. So for some reason, class 1 is evolving much higher diversity than class 2. And the question is why, right? So we know, you know, from a descriptive perspective that class 1s are more diverse, but I've always not been entirely sure why this is the case. And I don't think I fully understand after this paper either. But the interesting thing about this paper, and this is something that I think is something that you can plainly see, is that MHC evolves in apparent contradiction to species relatedness, right? So here on this right-hand side, you have the species tree, and it is roughly organized in Roy G. Biv rainbow colors, right? So HLA, that's up here, that's human, right? And then chimpanzee is our nearest relative, gorilla, orangutan, all the way down to, I don't even know what these guys are. Saki? Yeah, I don't know. Some other monkey, right? If you take a look at how we're related at a species level, this makes sense, right? We know our closest cousins are chimpanzees and bonobos. But when we look at our MHC genes, it's completely different, right? Like HLA, we're right here in the tree, and our nearest cousin looks like, what's green? Green looks like old world monkey, right? So even though our closest relative is the chimpanzee, when we look at our HLA genes, our MHC genes, our nearest neighbor is old world monkeys, which are pretty far from us in terms of our evolutionary relatedness. Does anybody have a hypothesis as to why this might be, this apparent contradiction? Does this strike people as bizarre? Because this is it's pretty weird to me, honestly. It is pretty weird to me. Give me a moment to hydrate. But I just want to stay on this for a second. This is very interesting, but how does it make sense? I don't know how it makes sense. We're about to find out what they did or what their arguments are. But when I first looked at this, I was also like, that seems wrong. Because if our closest relative should be a chimpanzee, then the left hand part of this figure, this HLA red box, should be close to a chimpanzee, so it should be close to pink. But pink isn't even on here, and gorillas are like way down here. So that's really weird. It's really bizarre. My best guess is difference in diversity pressure. Yeah. I think that's where my head went too. Like, there's something about how weird, and again, I keep on coming back to this idea. This protein is weird, right? This is, you just chalk it, throw it into the pile of weird things about this protein, right? It does a different thing in the immune system and in the nervous system. It has 10 to the 23 different combinations you can inherit them with. The relatedness to its closest species partners is quite different, and also it doesn't obey what we believe to be true about evolution, which is our proteins should be similar to our closest cousins, and this is kind of flying in the face of that. It's really, really bizarre. Our ancestors got freaky and traded with other species. I think that's true for certain aspects of our evolutionary history. Not entirely sure that counts for HLA, but you know, it is an interesting idea. And from that I mean primarily Neanderthal, right? Neanderthal interbreeding is like a thing. Is this something that can be easily toggled on or off while still being preserved? I'm not entirely sure to what aspect you are referring to. Okay, so what is their explanation here? MHC might be evolving too fast, or our records might be incomplete, right? We obviously prioritize sequencing human DNA because we're human and we're selfish, but there might just be gaps in the, you know, every year we discover hundreds and hundreds of more HLA variations. So the fact that we care mostly about humans and don't care quite as much as distantly related monkey cousins, this could be an artifact of us not fully understanding what those other proteins are like. But that's a little sad. It's a little sad to chalk this contradiction up to we didn't collect enough data from monkeys, which could be true, but it would be sad again. Kind of like how I think about panspermia, right? It could be true, but it would be sad. Okay. Have I missed the definition of how species tree were made? Here it looks like the sequence is credible while tree isn't tied with anything at the moment. The species tree, I'm pretty sure this is based off of genome-wide sequencing. I think that's how modern taxonomists do species relatedness, is through looking at how similar people's genomes are across their entire chromosome lengths. Could be wrong on that because I'm not a taxonomist. Maybe along the lines of the HLA mutates faster in humans than in other species. It's also possible, right? Like this, it's a weird protein. So one potential weird thing that could be happening that the authors propose is this thing called gene conversion. So chromosomes do all sorts of weird and wacky things. We actually referred to this earlier in chat when we talked about genetic linkage, right? So in bold, let's say you have dad's chromosome, the chromosome that you inherited from your dad, and in thin lines, let's say that's mom's chromosome, right? And so while your eggs and sperm are developing, there's a weird thing, a little glitch in the system that is not really a glitch, but it looks like a glitch, where you can have parts of the chromosome that look the same get scrambled. Right, so I've explained this a couple of times in my videos, so I'm hoping not to have to do a full explanation every time. But basically, if the DNA on one strand looks the same as the DNA on the other strand, they can get accidentally tangled up. And then when they get untangled, you can have genes do things like swap places. So this can cause scrambling of genetic sequences. So gene conversion happens when one gene sequence gets scrambled with another and then kind of pastes itself on top of another sequence. So this is like, they're saying that some of the weird genetic history between these genes is the fact that you could have like an ancient re-scramble a couple of times, that might make the taxonomical tree look not as we think it should because of these, very dramatic changes in genetic sequences. I don't really know if that's true. I kind of just nod my head and say, okay, I agree, I guess. But I'm gonna be real. I'm not enough of a evolutionary geneticist to like know how these ideas connect, but I figured I'd shout out their idea here. Yeah. So with perfect information, the two trees on the right should be pretty close to each other, but they're not. And when we put in all the data, so this is just the class 1s, I think class 1As, but when we put in all of the classic family members, you just get chaos. So when you do assemble all this data, it looks nothing like the species tree, and that's really confusing. And part of what the authors here want to do is to try and find a way to synthesize all of the information across the literature in a way that allows us to get a history of this protein that makes some kind of sense. Because right now, this makes no sense, right? It makes no sense that our HLA is closer to some random lemur than it is to gorillas. It wouldn't be that way in reality. Would this suggest that our understanding of human evolution is wrong? That is one takeaway that you could walk away from, but I don't think that's true. And we're going to see how the authors address this in just a little bit, right? Okay. Pseudogenes. We're back to the E. coli video. Pseudogenes. So these are genes that I talked about ad nauseum in that video. So pseudogenization is a process by which genes that were real genes at some point were mutated to the point where they are no longer real genes. And so people stop paying attention to them because they're not real genes. If they don't do anything, why would you study them? So a lot of attention gets taken away from regions of the genome that kind of sort of look like a real gene but aren't real genes. So I think the novelty of what the authors were contributing to the field is, okay, if we just take the data across the field, we get this tree that makes no gosh dang sense, right? It looks like MHCs violate some aspect of human evolution, but I think the way in which the authors provide a novel contribution is to also incorporate the analysis of these genes that look like genes but are not actually genes because they've been mutated and broken. And there are quite a few pseudogenes that are located in this region of the genome. So, as an example of how a pseudogene can occur, you have the very basic structure of a gene, which is a promoter and a gene. The promoter is a region of DNA that recruits RNA polymerase. Without RNA polymerase, you don't get mRNA made. Without mRNA, you don't get protein made. So a promoter serves to turn on a gene or enable a gene to be turned on. You could have a mutation in that promoter that stops the RNA polymerase from binding, ergo, therefore pseudogenizing that gene, making it no longer make a protein. So if you include pseudogenes in your analysis, now what does it look like? It's challenging. This is the turning on and off, which I meant earlier. I see. So when you include pseudogenes, look at what happens to the clustering. So it's straight up impossible to show these trees in detail. Like even the picture that they uploaded, when you download it and zoom in, it's really blurry. And I'm so sorry for my colorblind folk because it's going to be really annoying to tell what color is what. But what we're going to try to look at here is how well this story makes sense evolutionarily speaking. Because with all of these genes now in tow, when we do this historical analysis, we can now see whether or not we get clustering that makes some kind of sense. So if you are to look at these charts from far away, do you see greens segregating with greens? Do you see reds segregating with reds? Do you see blues segregating with blues? And on the left-hand side, we have exon 2 and exon 3 in the middle. These two are the parts of the MHCs that are known for binding to peptide. So these are going to display the highest variety of evolution, whereas exon 4 is the part of the protein that does not bind peptide. So that should be relatively stable across different species. So what do you see here? What do you see here? There's quite a distinct stack of green showing up. Yeah, it looks like using this level of analysis that you do get clustering. It doesn't look quite as neat as the like human on top, chimpanzee on bottom, but you do see that the greens tend to go with the greens, the blues tend to go with the blues, and where there is reds, there's also likely a neighboring red or a neighboring pink. A bit, but there's still a lot of stuff that doesn't match up. That's just science, right? It's not a complete picture. And again, for reasons that the authors had previously mentioned, we don't have a complete and robust database of primate MHCs. You could get weird things happening like gene conversion. There's other things that explain why this doesn't perfectly line up with the species tree. But there are clusters, right? That's an improvement. That is an improvement. It is neat. So what do they do with that tree? So I'm not a good enough evolutionary person to like walk you through that tree. I believe the important thing to look for is the clusters. But what they did at the end of this paper is this beautiful reproduction of the history of the MHC alpha block in the semiform. So this is the history of what we would call HLA in us, and then it's a different name for every other monkey, orangutan, gorilla out there. Right? This figure is quite pretty. It's really hard to tell what it is, so I'm going to zoom in and we can actually see the bubbling history of this crazy protein, right? And I think that's very neat. So at the beginning of time, who knows when this happened, but you had a protogene, so a gene that was before any of this history happened. It duplicated at some point, so you had two distinct versions of this, and you can see that this gene rather this segment of the gene, right? Because we're talking about segments. This segment of the MHC, what we would eventually call HLA, right? Sorry, I should rewind. I'm not 100% sure because the paper doesn't explain this, but I'm pretty sure these little blocks are these like little chunks of the HLA gene, right? Like little regions of the gene. So altogether, the whole thing is the gene, and then the little chunks are specific regions of that gene. I believe that's how this is interpreted, although I could be wrong. How they figure out so much history? You gotta ask an evolutionary biologist or a genetics person. I don't know how they assembled this. It was probably very difficult, but you can see how like I can't, like I was not able to do so for myself. Does MHC equal major histocompatibility complex? Yes, that is the topic for today. So yeah, you can see duplication, that whatever this portion of the gene was is duplicating and becoming its own unique thing. So evolution is tinkering with these, and then we have a split that is happening. So at some point in some animal that became the simians, there was a divergence where one of the versions got lost in some tree of primates, and then the other tree kept both versions, right? And then more duplications to this very complex gene, and then a region was deleted in one of the evolutions here, and I'll go into the next slide. So new regions get added in, duplicated and lost, and it's been traced. Exactly. And we look at the bottom, we can kind of see that, like, I don't know what this animal was all over here on the left, but in the gibbon, it looks like they lost a version and then had a huge chunk of that gene deleted to the point where the gibbon, as far as we know, has a very simple version of MHC, HLA-A. However, they do mention here that sequencing of more gibbon haplotypes is needed. They don't believe that this part of the tree is very accurate just because we don't have the data. Similar with the orangutan, that they need more data, need more orangutan haplotypes, but you can see that it is, I don't know, it makes me feel some kind of way to be able to see our shared evolutionary history. Like we have a kinship with these other simians, right? Our cousins, the chimpanzee. Between the two of us, they lost two versions of this gene, the haplotypes P and N, and we kept the one that's HLA-A as well as these other variations. But yeah, I wouldn't be able to completely walk you through these, but just seeing the visual, I think, is enough for me to be impressed that we can reconstruct the evolution of this rapidly evolving gene and use it as a way to relate to our primate cousins. And I think that's cool. I think that's cool. So, to sum, MHC, which are HLAs in humans, are a rapidly evolving immunological fingerprint that is highly variable across individuals to offer us some population level diversity. Even within the simians, MHC evolves so fast that it breaks the logic of evolutionary trees. But by adding all of our modern data with data on gene birth and death, a coherent history of MHC evolution across our simian family members has emerged. Now, it's not perfect, but the fact that a history can be assembled at all, I think, is really, really impressive, even though I don't understand all the aspects that go into research like this. I think it's pretty incredible. I think with this reconstructed tree, we can probably retrace the history of immunology that is built to our very own DNA. And that's it. That's it for the presentation. Let me move into the pond. We're not talking about glioblastoma, we're just chatting. That's it for the presentation. How was that? What was your reaction to learning about MHC, my favorite protein? Is it your favorite protein too? Have I maybe convinced some of you to adapt MHC as your new favorite protein? I just love this thing so much. I did have a different stream where I talked about this protein, but I did not, that's why I delay videos until I feel like I'm ready for them, because I don't want to squander my one shot at communicating a topic that I really love, right? Like glioblastoma, I had the idea to cover it, but I wasn't ready for a long time. And then I felt ready because I felt like I knew exactly how I wanted to tell the story, and I don't tell stories until I'm ready to tell them, essentially. So it'll be a while because, again, you can tell the degree by which I love this protein is quite high, and therefore it must be treated with a lot of respect. Not perfectionism, right? I'm not a perfectionist. Like, let me make this clear, because I think I got accused of being a perfectionist on my last chatting stream. I'm not a perfectionist. I will be happy by doing the best job I can do given my current circumstances. And I feel like, given my current skill set and storytelling ability, that I don't have it yet. I don't have the nuance, the storytelling, the editing, I don't feel like I'm ready yet, but I will be one day. That's what separates me from a perfectionist, is I'm gonna do it. I'm not gonna let my need for everything to be the best thing that I've ever done get in the way of actually covering it. It's just I will wait a while. Yeah. Phylogeny haunt me again. Yeah. Yeah. I said that this was me like brushing my teeth. It is brushing my teeth as someone who does not want to brush their teeth. I do brush my teeth, obviously, but in terms of like, eating my fiber and brushing my teeth and doing all the exercise, like trying to read papers that are not in my direct expertise is keeping me sharp. Which is especially important for me now that I'm no longer teaching at university, that I always have this feeling that I'm losing my intellectual edge. So, there's this weird pressure of like, I need to make these videos while I'm smart, because I can feel the smartness leaving, because I'm not being challenged by other people anymore. I have to constantly challenge myself, which is harder to do. It's more active. Like, you take more of that responsibility, which is, sometimes you just want to play Slay the Spire, right? Like, you don't always want to challenge yourself. Interesting. It's been a protein of interest. Love learning more about it. That's awesome. That's all I want from this. I appreciate the work that goes into these. Thank you. I'm glad that the work is recognized. Mind blown, isn't it? It's awesome. P53 is awesome too. Oh yeah, P53. I mean, I feel no need to talk about it because I talk about it every time cancer comes up, which is going to happen at least once or twice a year, right? For the charity streams, as well as the delayed full video that I make on certain cancers. It's my favorite protein now. That's awesome. I'm glad. It should be everyone's favorite protein. I love the stupid protein. It's good. It was a little difficult to follow. The thread was fascinating nonetheless. Yeah, especially this last part, I feel like didn't really have a thread, but that's really on me, because I don't have the skill set to understand this paper fully. My brain is going to be buzzing with this for a while. Thank you. Yes. let me tell you, when my advisor told me the story about how different viruses subvert MHC, that's a great way of putting it. Like my brain was buzzing. I like couldn't wait to read the paper that she was referring to. Like I couldn't wait to do my own research and actually, crawl the literature to find out, how clever these viruses can be. Oh, by the way, I know. I said I was only gonna allow myself one like comment crash out a month, but like I had a comment that was on my Ebola video where it was like, this guy is making a big deal out of nothing. All viruses usurp the immune system. Ebola is not special in how it invades the immune system. Don't listen to this guy. This guy's all hype. I'm like, bro, that's the point. The cool thing is that every virus has anti-immune adaptations. What do you mean it's not interesting? Of course it's interesting that every virus has its own unique special snowflake way of subverting the immune system, and no two viruses are completely alike. I just got that's such a midwit. I really wanted to avoid using the term midwit, but I think I like the term enough, because the way that I'm defining midwit is somebody who denigrates other people's intellectual efforts in an attempt to themselves appear smarter, right? What does that commenter get out of insulting my video, calling it like overrated, that I'm excited about the unique evolutionary adaptation of Ebola to our immune system? Like there's no actual living scientist on planet Earth who studies biology in a professional capacity that would ever be like, well, Ebola's not that cool. Like that comment can only come from somebody, I think, who has this like midweight complex. I think it's crazy to get that kind of comment. I promised I would only allow myself one of those, but that comment just was so baffling to me that like there could be a human out there that looks at the effort that I put in these videos and goes, it's not that cool. I'm not that impressed. Oh, it's crazy, dude. It's crazy. No, it's crazy. It's not my favorite yet, but I was so happy to learn all of this. I'm curious, what is your favorite protein then? What is your favorite protein then? Out of curiosity. I think one can be forgiven for thinking twice before like streaming live on the internet. I no longer know what that's in reference to. Sorry, forgot the context. Oh, like my coverage not being perfect. Yeah, but I make sure to like hedge a lot. Love the heating prevention proteins, because if they didn't exist where I live, I would be a pond. That's fun. That's fun. Currently working on my master's in math. I want to do a 180 into biology/math biology. I currently work in biomedical field at SNPRC. Have you seen a significant math modeling research in mole bio? Yeah, math and programming and mole bio are very intertwined, right? There's genomics, there's every omics you can imagine will require some level of math, statistics, calculations, and they're also really important and somewhat robust to AI taking every job, I think, because you still need a human to interpret omics data, to quality control omics data, et cetera, et cetera. Phylum wept because there were no more proteins to study. Nonsense. There are so many cool proteins out there. MHC is just my personal favorite because I have a deep history with this protein. Me and MHC go way back. I feel that I finished my OChem and I feel it fading as I read papers on synthesis. It sucks because I'm like, I think my peak critical thinking, my peak analysis was like maybe 2 years after dropping out of my PhD. And then from there, I can feel my ability to engage deeply with a paper to be slipping as I get less and less familiar with the nuances. I think I'm a better critical thinker than I ever was in my PhD. But I think I am getting less familiar with the stuff, the experimental nuance, which sucks. There's not a whole lot I can do about that other than go back to school, I guess. A personal hypothesis, I think cells evolve along two axes, free-flowing cells that don't belong to a system. that want to spread and survive, like viruses or bacteria, so they evolve in anti-MHC direction. And two, cells within a system that want to cooperate and keep the whole body alive, so they evolve in a pro-MHC direction. I think that's pretty interesting. Interesting how you would react to the weirdo lamprey immune system that doesn't really do MHC stuff. It is really neat. Some people are so incurious. It's crazy, right? Like the person that needs to say something like this video was not interesting because all viruses do this is a very sad person. It makes me sad to know that that human exists. Whenever I respond to these comments, it's never one out of like hatred or malice. It is, your life is such a way where you are clawing back a sense of self-importance by dunking on my self-importance. Which is just shocking to me that there are people like that. Yeah. Being ignorant is often okay. Weaponizing your ignorance is not okay. I agree with that. It's so funny to use like, and they're not wrong, right? Ebola is not unique in the sense that all viruses do usurp the immune system. Ebola has certain things that are unique about it that make it different than other viruses. So if you wanted to say viruses aren't interesting because all viruses do the same things, that's a legal thing that you can say. That's not a wrong thing you can say. I just think you have bad taste. I think you have bad taste if that's what you say. I'm working on heat shock now. That's awesome. I think heat shock proteins are really interesting. So heat shock proteins are named such that they are proteins that undergo a conformational change, meaning they change shape in response to a sudden burst of heat, a rapid change in temperature. And so that makes them really good at coordinating a anti-stress response. If we're talking about like yeast are famous for their heat shock proteins. Although I don't really know a whole lot about heat shock in humans because we're usually pretty good at staying within a certain physiological range. So I do know that heat shock proteins in humans are important for protein folding, but I don't actually know too much about how they are regulated. A bit of a blind spot of mine. Ferritin is mine. Oh, I don't know a lot about ferritin. I'm guessing that's just a protein that binds iron, but I'm guessing if it's your favorite, it's probably got something else to it. It looks like a Dyson sphere. Oh, it's that one. I think I've seen it before. That is cool. I don't actually know about proteins. This is why I was so happy to learn about one. Well, I'm glad to be able to have one of the first proteins you learn be one of my favorites. Vault protein, Vault video when? You know, maybe you'll see some Vault related content on your feeds at some point. I'm not saying anything, but I'm just saying, well, a Vault is very, very interesting. Very, very interesting. A huge, mysterious immune complex. You know, I'm not promising anything, but I'm saying that maybe in the future you will see the Vault on your algorithm at some point. Yeah. I think it'll change because there's so many. I've heard that some people are custom designing proteins. Absolutely, with CRISPR. CRISPR has made it so easy for any researcher out there to just make up their own proteins. Designer proteins that can be used to do all sorts of interesting drug interactions. Really, really cool and interesting engineering applications for designing your own proteins. AlphaFold helps us predict protein 3D structure, a huge jump. Absolutely. Love AlphaFold. Used AlphaFold for this presentation. What do you think about connecting machine learning models to biological organisms like rats' brains and reading their minds? There's a paper in the Infinite Backlog that literally reconstructs a rodent's vision, like from their brainwaves. Like you can now, in neuroscience, you can reconstruct the perception of vision just from data that you get off of neurons in a mouse, which is insane. So how do I feel about it? I feel that it's cool and very useful. Honestly, if things like AlphaFold were what we used AI for, nobody would have a problem with it. Exactly. That's why I'm always careful to say I'm not anti-AI. I'm anti-AI replacing art. And I've repeated that multiple times in multiple different videos. Other than that, TFID, EF2, the entire nuclear polar, glycodelin, I don't know that one, clathrin. I do like clathrin. Ubiquitin all come to mind. Great proteins. Yeah. It's very weird. That might have been a comment out of bitterness, actually. I once had depression. If I saw that at a time, a video of someone having this much fun over something, I can't anymore. Oh, it could be that. I mean, it still comes with the same feeling of like, I'm sorry your life is like this. Like, yeah, I mean, it might not be anything that I did, but any video of anybody having fun would have been a bad thing for them, which is also why, like, to encourage other people to go out there and post your content to places, a lot of negative comments I get are from places that are like this, where they're not usually like legitimate criticisms of my content, but it's like, man, somebody had a bad day and they decided to take it out on me for some reason, which is, sad in terms of relating to another human being. Do you think the relations between pro-MHC expression has pushed many people to have autoimmunity problems? I don't know enough about the role of MHC in autoimmunity, to be honest. So I'm not going to comment on that other than to say that's an interesting and excellent question. Do you think that's cool? They found something better than Tiger, than CRISPR-Cas9, Tiger TAS. I did see Tiger TAS. I don't know if/when I will cover that. Seems more like a news channel thing than a deep dive molecular biology thing because it is so new. I'm not taking it off the table, but I am saying, don't know if I will. Not yet. My favorite protein is Rubisco, just because it's so silly in an evolutionary way. So for those of you who don't know, Rubisco is a crucial carbon fixation protein that plants have, and I think one of the engineering problems that would result in the salvation of humanity is to improve Rubisco just by a little bit, because it is the only thing that we know that can do its job, which is to help photosynthesis. And if we could get more photosynthesis to happen, the world would be a better place. Because of the excesses of capitalism, our world is being irrevocably changed, and Rubisco is one way where we could potentially meaningfully undo damage to our planet, as long as we can get the engineers to actually crack that code. Yeah, Rubisco is super cool. I don't know what you mean by silly in an evolutionary way, other than the fact that it is pretty inefficient at doing its job, but it's the only thing that we know that can do its job, as far as I can tell. Yeah. That was a cool paper, the rat brain one. Yeah, it was cool. It's cool. There's a difference between research AI and generative AI. These are fundamentally different. Yeah. Unfortunately, they do have the same. They take up the same mind space in the public. So it's really nice to have a space in an audience who recognizes nuance. And so I'm glad to be preaching to the choir, essentially. Rubisco is a wild choice. It's a good choice if you like chaos and defying destiny. I think Rubisco is an excellent choice for a favorite protein. So you want more creators to post and soak up the sad person if he's keeping your content safe. Genius. Well, my mental health is not actually negatively affected by these comments. It is actually good for my empathy, I think. There's that channel, Financial Audit, with Caleb Hammer, that's all about like making people's bad financial situations into exploitable spectacle content. And at first when I was watching it, so I was like, oh man, people have made some very bad financial decisions. But I stopped watching the channel because I could feel my empathy for my fellow human being be irreversibly drained. Like, I started feeling angrier, and engaging content that is algorithmically friendly is designed to make you angry and binge and hate watch and everything. But I recognize that, like, watching these people do bad finances or have bad situations done to them, but in a medium in which I am led to place blame on the individual for their financial choices and decisions, regardless of whether or not you want to say that's right or wrong. I don't care if you think it's right or wrong. What I am saying is that the undeniable fact of the matter is that I felt my empathy go away as I was watching the channel. And then comments that are like this where it's like I could get really mad at this person and be like, what the heck is this guy's deal? But it is useful for me to instead take that person's perspective and put myself in their shoes, not as a way to excuse their behavior, but just to realize that, like, something in this guy's head is different than in my head. So in the very basic task of empathizing with another human, not to say that they're right or that I feel sorry for them, but just to be like, what had to have happened in this person's life for them to leave this comment is a useful exercise in empathy, I think. Now, I don't feel warmly to that commenter, but I do feel like cosmically sad. Cosmically sad for that commenter. Yeah. Tiger Task doesn't require PAM, and it can theoretically target any genomic site. It's small, so the delivery could be better. That might be fine for a journal club. I don't think I would make a video on it because that's like more of a, I don't know, Hank Green, Veritasium, Kierkegaard kind of, this is the newest thing in science, whereas I prefer to like look into the past instead of cover what's present. Online spaces make us better. Not all of them do, right? Like, I think my community here is quite positive. The streaming community, y'all care enough about this science and care a lot about the biology and enough about me as a creator to show up to these, so that I am appreciative of. It does feel very soul destroying. Not gonna lie, you have a very healthy way of dealing with hate comments. It helps that I don't get a lot of them. Right. I mention them occasionally just to point out that, like, it is the exception and not the rule, but it is normal to get hate comments, which is different, right? Like, if you make any kind of content out there, it could be the most inoffensive, rationally good piece of media, and you're still gonna have, because humans are very different, right? Like, I think doing YouTube has made me fully appreciate the diversity of lived human experiences, where, like, I can put it sounds backhanded, but it's actually true though, where like I make an hour-long video and I pour my heart and soul into it, and then somebody like gets obsessed over like one minor mistake and then writes a paragraph of that one minor mistake and then calls me someone who spreads misinformation. Like, that's a person I didn't know that existed, because in, for example, a biogenesis video, I do a lot of hedging. I do a lot of explicit, like, make up your own mind. I'm presenting the evidence, the strengths and weaknesses, and I get a lot of comments that are like, you're biased, you're pushing an agenda, where I'm like, I'm literally saying, I'm comparing the models and telling you that I have a bias. It's true, I have a bias. And then the fact that there are people who ignore all of that and then leave a hate comment, I'm like, wow, we are really different. And I have to remember that on the spectrum of humanity, that you, the hate commenter, also exist. That we technically share the same species, even though my existing mindset is pretty different than yours. And to maintain a sense of empathy, I have to remember that you also exist. Crazy. This video will make you angry by CPG Grey. That's a great video. That's a video that really stuck with me. The like anger dopamine reinforcement loop. But I don't want to do that, because that's actually soul crushing. What I want to do is make fun content that I have fun doing. And then the engagement can come from me baiting people with essays that show up on the screen for like 10 frames instead of this like rage bait, which is so... It's also exhausting to be like accused of rage bait, which is just untrue, because like, I can admit because I've pinned comments before that have corrected me when I've gotten things wrong, but to be accused of active malice is also like, dang, you are also a person where I think I have constructed a very calm and agendaless video that is just about the edge of research, and I have been accused of pushing an agenda. I have to remember that you as a human exist too. Like, I don't know. It's a weird exercise that I force myself to do. You thrive in a niche topic, go against the meta. Yeah, I'm like, what is it, like R versus K selection in terms of evolutionary strategies. I'm less of a bacteria and more of an archaea. Right? I thrive in the hot, bubbling hot springs of YouTube audiences who really want to think a lot about biology. A niche that I didn't think existed, right? Because there's not a whole lot of popular creators who are doing this, so there's not really a sense for how many people like you are out there. But I'm happy that you are here with me. There's a SciShow vid about Tiger Task. I figured, right? Because their coverage is more about recent advancements in science than mine is. Because I don't think I could be. The thing is that for me, in order for me to make content, I need to be really excited about it, which takes time. Tiger Task is relatively new, and I haven't had the time to fully explore the full story in the way that I experience it. Yeah. A fascination with emergent complexity definitely acts on a person in a way to make them more focused on casual analysis over moralization or whatever else. I'm not entirely sure what you mean by that. Both have value, but I really enjoyed diving deep into a polished subject. Yeah, both have value. I'm not saying that it's above me to like cover new topics. It's just I could not do it better than Hank Green can. Right, I'm not capable of doing it better than Hank Green can. I'm just me and my wife, so that's also why I can't do it better than Hank Green can. But that's like that's how I think of myself, as like an archaea, like a R selection, K selection, archaea. Like I don't want to compete with Kierkegaard or SciShow or CPG Grey or anybody. I want to find my weird niche and convince you that the niche should exist and bring people into that new niche. I think that's a much more interesting, sustainable option. I think it's cool. From what I know, you are the main channels for Mobile, me and Clockwork. There's also, oh crap, what's his name? Nano Rooms. Nano Rooms. If I have another channel recommendation to make, there's Nano Rooms as well. Nano Rooms, go check them out. Trying to think of other creators in this space, but yeah, not a lot that do what we do. Specifically, there's like a very deep dive into molecular biology. We're not the only biology channels out there, right? But if you want like cell and molecular stuff broken down for the average person who is interested in this stuff to consume, I can't think of another person who's doing that. I would like to hear more about your research process for your vids. So it starts with a thumbnail. I usually will only, well, nowadays I will only agree to do a video if I can think of a fun thumbnail / title to build the story around, because I mean, come on, it's YouTube. I got to think about the YouTube meta, right? So it starts with an interesting thumbnail idea. So even though I'm really interested in, for example, the fact that plants have been surprisingly tolerant of whole genome duplication, right? So if you haven't watched my new gene video, here's a fun fact. Humans have, oh gosh, I don't remember any of the numbers. Like 300,000 genes, something like that. And then like a onion has like millions or something. It's like some ridiculous difference between the amount of DNA in a single tomato or a single tobacco plant cell. Like they just have so much more DNA than humans do, even though we count ourselves as quote unquote more complex. And so what follows is, well, why do they have so much DNA? And a lot of them have a lot of whole genome duplications. Like, we're diploid. We have one copy we get from mom, one copy we get from dad. But like, I forget exactly what the plant was. Like the tobacco plant, I think, has like 4 copies. There's like a fern that has 8 copies of their genome. And in the majority of vertebrate cells, animal cells even, if you, oops, all chromosomes, if you accidentally duplicate your chromosomes, that cell is usually dead. There are rare exceptions though, right? Like there are a whole class of diseases called aneuploidy, where you get the incorrect number of chromosomes in the sperm/egg, which becomes a zygote with the incorrect number of chromosomes, and those individuals have physiological problems, put it that way. But on a cellular level, usually if you accidentally oopsied an extra or not enough chromosomes, the cell has apoptosis, it'll blow itself up, it'll prevent that error from propagating. Even if it could potentially, hypothetically, survive, there's a cellular program to trim the cells that do not operate at a certain threshold of correctness, we'll say. So a video that I've wanted to do for a long time is, well, how the heck does a fern have 8 copies of every gene? It just seems like apoptosis would have destroyed that possibility a long time ago. And the problem with that video idea is I can't really think of a good thumbnail right now. So the process starts with a thumbnail, but it actually starts with a lot of thinking before that, right? Like I have to identify my personal feelings with regards to the subject. Like it has to make me feel a certain way, and that feeling gets converted into the thumbnail. So for the glioblastoma video, you know, I covered glioblastoma 2 years ago or a year and a half ago or whatever, in that 24-hour, 12-hour charity stream. And the feeling I got when I did research for that video was one of this cancer is like taking over my brain. It's grabbing these neuronal connections. And there was a very uncomfortable feeling that I wanted to convey in the story of that video. So before the first line of a script even gets written, I first have to identify what about this makes me feel a certain way. For the genome duplication thing, the fern thing, it makes me feel like there's like a glitch in the system, uncomfortable, uneasy, because biology is not functioning as intended. This fern got away with one too many too many duplications, honestly. Yeah, so it starts with a thumbnail. It starts with an emotional connection to the research topic. I mentioned this on my last chatting stream, but the only video I don't have an emotional connection to is my kombucha video. Everything else started in my brain as an emotion, a compelling emotion about the story. If it's just an intellectually interesting idea, I don't do it until I can find my emotional reaction to the information. Yeah. Bro, talking about niche topic, talk about Yamanaka factors. Don't remember what those are, or induced pluripotent stem cells. That I could probably do as a journal club at some point. Humans have 20,000 genes. Ah, that's right. Yeah, that number I was referring to is base pairs. Sorry. I remember hearing somewhere about carnivorous plants evolving possibly through whole genome duplication. Yeah, it sounds really interesting. I just can't think of a way to tell it. Humans occasionally have an extra chromosome with some more or less apparent effects depending on which extra chromosome. Why does each duplicated chromosome has the effect it has? Well, it's just chromosomes have different genes on them, right? So a little extra this, a little extra that, it's going to differ between which chromosome gets duplicated. That's about all. That's the level by which I understand aneuploidy, at least. Is this common or rare in other animals? I don't have any expertise in other animals. My expertise is primarily mice and humans, and then to a lesser degree, yeast, bacteria, flies, worms, and one fish. 416 chromosomes. Yeah, it's crazy. It's crazy. I don't know about a title, but you could put a mountainous amount of DNA for the onion with a human one for scale. Yeah, I think that would be a pretty cool thumbnail. That's not a bad idea. But I just got to know as a neutrophil, what do you attribute your freakishly long longevity? I'm the like 1000th fly or something. Every time you watch a new stream, every time you see me appear in a video, it's actually a new phi. Because the average lifespan of a neutrophil is 24 hours. So I'm just, after this stream, I will expire and be replaced by a new freshly differentiated phi, the neutrophil. That's how that works. Right, so then, what comes after that? Oh, I'm so deep into the pond, I didn't even realize. What comes next after like thinking about a topic and having that emotional connection is that I script or I outline. I do either or. If the story I'm willing to tell is like very not that complicated, I'll just skip the outline. And if it is complicated, then I'll go through an outline first, because the outline helps if I have a lot of complicated ideas that need organization. So, for example, glioblastoma, there's all sorts of different topics within glioblastoma that I could talk about, and an outline really helps me determine which ideas I thought were good enough to put in a video and which ideas I thought were just a waste of time. For example, I had a portion of the glioblastoma video talk about the immune privilege of the brain, which was going to take like another 7 minutes. I was just like, for a stream audience, I think it's interesting. For a video audience, I just don't think it was worth the minutes of additional production. And then from the outline or raw, I'll just go into a script and talk about the topic like I'm telling a story. And of course, there's a lot of rewriting that happens between drafts. I read out loud a lot. That's part of my process. Is I don't just read in my head. A lot of the rewriting comes from hearing the way that my words come off, because I care a lot about how I sound, because I have like a little history with poetry and performance. I did a lot of acting as a kid, and like monologuing, and I like theater and stuff like that. So there's a lot of listening back to the theatrics of my script. So that's the outline of how I write a video. Isn't the main thing about pluripodia in plants that crops with larger genomes tend to produce more? But how do you balance that with the reduced fitness of having to take care of that entire genome? That's what's so curious to me. Is that the longest live stream is 24 hours? Exactly. Exactly. Yeah. Thank you so much for the 10 joules. Didn't really appear on my screen, but I'll take your word for it. I don't even know what joules are. YouTube's doing all sorts of fun things. I really like how your videos are stories. Thank you. That's an important part of the process to me. And that actually comes from a background of being bad at biology, if you could believe it. Because I know some of my pre-med friends, they could just bash things into their head. You just give them the notes and they'll just bash them into their head, do well on the test, and then move on from their lives, right? But I couldn't do it because I've talked about this multiple times on stream. My memory is actually really bad. Not only long-term, short-term, working, all of my memories are really terrible. And so the skill that you get from being bad at memory is coming up with ways to patch up your weaknesses, right? So that's why, I always like to talk people up. Part of my job as a teacher was to make sure my students had good self-confidence, is that even when you are disadvantaged in certain ways, for example, I don't have like a clinically diagnosable memory defect, right? But I believe I test below average for somebody, right? And I think there's a lot of doomerism when it comes to life has dealt me this particular hand, and I can't do anything about it. To a certain degree, that's true, but at the same time, like, by overcoming the bad cards in your hand, you actually get additional benefits. So my poor memory allowed me to teach better because I could better empathize with students who are also similarly bad at memory. And so how do you effectively teach a student whose memory is bad? Well, instead of providing them with a list of disorganized facts, you provide them with a story. Stories are a lot more interesting, more engaging, more memorable than just lecture slides, right? So I appreciate that my care in telling stories is being appreciated, right? That means this is proof that when you overcome, or not overcome, because I'll never overcome my bad memory. That's just not possible. I'm biologically determined to have a bad memory. But in fighting that bad memory, I have developed a new skill, which I think is quite useful. And I think I can go for the same for a lot of people who have perceived self-worth issues, put it that way. Yeah. ADHD, working memory, counts as executive function. I'm completely neurotypical. I just have bad memory. I actually talked a little bit about this in my last stream where it surprises people to learn that I am neurotypical, but I feel like it's it's like it's complicated, right? Not whether I'm neurotypical. That's actually very uncomplicated. But my feelings about uh, uh, the perception that I am neuroatypical, I find that to be kind of funny. I find it funny because every time I test myself for autism, I'm like the most neurotypical person. Like I'm scoring extremely high in neurotypicality. I think the surprise comes from my communication style, which is, um, as you can tell, I take a lot of time to think exactly about what words I want to say, um, which I think, I mean, you can tell me if that's true, right? Because I don't actually know. Because to me, this is just the way I talk, right? I'm not like masking or anything. This is just how I learned to deal with certain obstacles in my life, is to evolve a very, I think I'm pretty verbose. I use a lot of words when I don't have to, but I'm also always listening for multiple ways in which my words can be interpreted because I just like the experience of saying something and knowing that I am being understood in a specific kind of way. That's it's just inherently satisfying for me. I would like a dark, soundproof room for just talking the stuff I'm studying with my own words, see if I have the faintest clue what I'm doing. I'd get into tutoring or something, you know, a class that you did well in. When you force yourself to verbalize your thoughts, you are engaging more of your brain, right? You're just working harder to do, which is why ChatGPT is so corrosive for learning, right? In some instances, it can be helpful to find information. The problem with ChatGPT is that it chews up the information for you and makes it maximally digestible. If you don't work to digest the complicated stuff that other people do, you end up losing some cognitive edge. I think that's also supported by research, where even as a study tool, ChatGPT can be corrosive. So that's why I say that, like, even when you have bad professors still teach you something valuable, and it's how to study on your own. Um, I hear stories all the time on stream of like, you know, my professor's so bad. I wish my professor were more like you. I'm like, I don't think that I can say that that is true. I think that good and bad professors both engage you in different ways. A good professor will keep you motivated. A bad professor will teach you to be self-sufficient. And I've had my share of both, right? But because I have had people fail to teach me, I have become better at teaching myself. The problem with ChatGPT is that it is good at teaching you, which means you don't have to learn how to teach yourself. And that's pretty dangerous. I guess some weaknesses force you to work around them. Although I imagine in university you're with people who have been selected for good academics. You'd be surprised. As a teacher, there are quite a few students every year who have extra accommodations for learning stuff. I don't want to call it a disability, but some people just need more time. There's nothing disabling about that, I think. I don't like referring to that as disability. It's just your mind works a bit different. There's no reason why you wouldn't be afforded extra time in the real world when it comes to recalling information, right? Like, there are certain jobs where neurotypicality might be preferred, but I think in the majority of jobs, it's fine as long as you are doing the work that is required of you. So in terms of good academics, yes, university can select for good academics. That doesn't necessarily mean that they select for people who don't have challenges to their learning, right? It's selected for people who may, you know, some people don't have challenges. They just are naturally very smart, and can absorb a lot of information, don't have to work too hard. I am envious, but if I were like that, I wouldn't be doing this channel. It would completely change my career direction. It is because I have studied and like struggled in school that I have developed a skill set that allows me to storytell. Because otherwise, why would I bother practicing storytelling if I didn't need to teach myself how to survive school, right? So I would say academia selects for people who have good academics, but that includes people who have challenges, because overcoming challenges can teach you about discipline, rigor, whatever you want to call it, right? That's like, I wouldn't think of it as a weakness. I just think of it as one additional difficulty level, but then because you are playing at a higher difficulty level, you actually get better at the game. Does that kind of make sense? I'm trying to be very encouraging, right? Like I don't think of these as debuffs or whatever. They're making you play at a slightly harder difficulty, but that means you can get better at the game of life, game of academics, etc., etc. Yeah. Speaking of memory, an interesting protein is PKZs, a zeta isoform of PKC, and its interaction with zeta inhibitory peptide. Those are words that I heard at some point in graduate school, but I've completely forgotten. I bet they are interesting. My memory used to be pretty good, but thanks to my mental illness and burnout, it's been nerfed, so that I'm trying to relearn how to learn. I think like part of like the philosophy that I abide to is accepting that these things happen. Not to, you know, I'm not downplaying the severity of what you were talking about, but I'm accepting that it has happened to you, and I'm also respecting the grind to relearning how to learn. It's not easy, but it is important. Talking slow is good for clarity. I talk way too quick. I actually made the effort to slow down in my last 2 videos. But I swear to God, you literally speak, teach, and think everything like I do. Well, we might have had similar upbringings. How you express yourself very clearly, and I appreciate that. Thank you, thank you. I think what I worry about in an era with LLMs is that the intrinsic desire to be understood is, you know, and if you watched my members video, you already know my thoughts about AI and art and human connection. But if you rely on AI for connecting with people, you know, AI, help me write a work email. AI, help me apologize to my girlfriend, boyfriend, partner. AI, help me write a best man speech. Yeah, I think the average person is not that great at communicating, and the AI could probably do a better job objectively. Like, most people, their best man speeches are gonna be terrible, too awkward, not funny, take too long. ChatGPT could probably write you an optimal best man speech. If you measured the quality of a best man speech by humor, time, length, you know, cleverness. But if you measure the quality of communication by connection with other people, then AI cannot help you communicate who you are as a human, right? So my background in technical writing, my background in grant writing, that has helped me with clear communication. But I think what is something that I want people to do more of is communicate to humans, right? Practice that. Practice communicating your feelings. That's always helpful outside of academia, right? To verbalize and express exactly what is on your mind. And that to me is like the greatest challenge, is communicating your own feelings, because how do you accurately capture emotion when emotion itself is irrational, illogical, of the biology of the brain? And by wrestling with those seemingly illogical feelings, I think you can really craft a way to better communicate. This is unsolicited advice, but I do care a lot about communication. Like, you can tell that, like, I think a lot. And I actually think that, like, the next stream that I might do might be a while, but the next stream that I want to do is like a rhetorical analysis of Kierkegaard, of Tirzu, of The Octopus Lady, of SciShow, of Clockwork, you know, some of my favorite science content creators, and actually break down what they are doing from my perspective as someone who thinks all day and night about communication, about art, etc., etc. You know, I did a little bit of it in my grant writing, my technical writing workshop as a stream, but like what I want to do is I want to sit and experience the art with you and then talk about what I think is working about the art. Because, you know, I've had people tell me that they like the way I communicate, but no curiosity as to how I got there. And I think it's meaningful to me that I feel like I am doing something that improves people's skill in some kind of way. Because with the biology stuff, like, okay, if you're a bio student or a med student and you're watching my stuff, I don't consider these resources, right? Like, let's be real. If you're a medical student, you're probably not watching my videos to study. You might watch my videos to stay motivated, but you're not watching my videos to study because I'm not covering the MCAT. I'm not covering Physiology 101. I'm not doing the things that you would need in a proper medical education, but my primary goal is to keep you motivated, to tell you interesting stories, to make you curious, engaged. Communication is something I could actually teach you, I think. I might sound haughty or arrogant for that, but I trust that if you think that you would like to pick up some of what I do, not in the way that I do it, because you're a unique human being and the way that you're going to communicate is way different than mine, but by looking at communication through an analytical lens like I do, like my kind of kung fu, whatever you want to call it, self-cultivation of like trying to get better at communicating. If I give you that framework, you're not going to develop into me. You're going to see what I see when I think about getting better at being me, and then maybe get better yourself. I think that would be pretty cool. Yeah. I really don't like to overdiagnose, but have you seen Hank's video on autism? He now tests as neurotypical, whereas he used to absolutely be not. I'm sorry, it's just so uncanny. It's uncanny that I'm like neurotypical. The way that I see it is I don't have any sensory anything, right? All the entire questionnaire on the autism questionnaire that I get, that's all about sensation. I don't have any of that. I'm just awkward, and I really care about my words, and I really like biology because I don't get obsessed about this over random things, right? I guess there are a couple of, you could call them hyperfixations, but I don't want to use that because I'm not neurodiverse, so I will just call them passions. Biology is a big passion of mine. The art of communication is another big passion of mine. The study of loneliness and the human condition is another passion of mine. And then all the philosophies that go into the study of loneliness, those are all big passions of mine. And V theorem. For those of you who don't know, I'm a part of a VTuber group who does STEM communication. Let's do a shout out to them. I'm also very passionate about developing an audience for SciComm content in the VTubing sphere. So if you like what I do as a VTuber, you can go check them out as well. Yeah. Truly horrifying how quickly the thought replacement is going. Yeah, ChatGPT just takes a load off of you, and you can use it. And like, I'm not anti AI tools. I'm urging people to be very careful with how they use them, because in my last members video, every time you choose to make something more inconvenient, or every time you choose to make something that's inconvenient more convenient, you are sacrificing something. And that something is literally the labor, right? By making something convenient, you are removing labor from the equation. Sometimes you can get away with that and it's not a problem, right? Like, we don't write letters anymore. I don't think people care that we don't write letters anymore. Like, why pay for postage when you're already paying for data, right? But that comes at a cost, right? Letters are personal. You can read Darwin's letters to his wife. They're wonderful. They're very heartwarming. They're lovely. I can imagine in my head, when I'm empathizing with these two historical figures, Darwin and his wife, how happy they must have been, how worried they must have been for each other when they talked about health issues, right? These letters are artifacts from history, but they do have a lot of meaning behind them. And it's just, every time you make something more convenient, you have to think about in ways where you can cling on to the things that matter to you. So instead of writing letters, you got to respond to that group chat, man. Don't leave that group chat hanging. You got to make sure you keep up your connections with people. Yeah. I value the flaws in communication that each individual has become. It means I know it's from them and not an LLM. Oh, dude, the discourse on flaws, I think, is going to be the next hot thing, the next hot commodity within content, I think, because I've talked about this show a lot, Has Been Hotel, famed show that people love to hate. I love the show. It's also very flawed. But I love it in part because it is extremely flawed, because it is like one person's Tumblr addiction became a real show and it's corny and cheesy, and it's got a lot of heart and energy behind it. And is it as uncontroversially great as Avatar: The Last Airbender? No, I think Avatar: The Last Airbender is like an artistically essentially perfect thing that everybody can agree is pretty much perfect. Like the whole culture, we all agree this is a good show. But I think inspecting the ways in which people disagree over a piece of medium is actually also very interesting. So I no longer look at reviews, because much like how I get hate comments and I don't think I deserve them, I think a lot of other art gets hate comments that don't deserve them, because Has Been Hotel is making a very strong artistic statement coming from the mind of one person, primarily, right? An artistic vision that is so personal, and that's just not going to vibe with a lot of people, because it is so individual in its expression. Whereas I think Avatar: The Last Airbender is the work primarily, the creative vision of two people, but there's a whole team that is crafting it very carefully to be as widely appealing and gripping as possible. Whereas Has Been really feels like they were going for a very specific audience, and if you're not in that audience, it doesn't really do anything for you. But for that audience, it goes so hard, right? And that's the same with my channel. I feel a kinship in a kind of way, because I'm not Kierkegaard, I'm not SciShow, I'm not widely appealing. I'm selected for in a very careful niche of people who really want to deep dive in biology. So this is going to go hard for some people, and it's not going to really be a thing for most people, put it that way. At this rate, AI is going to do all the science and poetry. That's kind of lame. I agree. We have brains. It's fun to do stuff. Write personal essays. That's how I got my start. Technical writing begins with an exploration of personal essays. What's the Octopus Lady? Go look them up. Fantastic creator. They do marine biology. Love their content. They're really, really funny and very, very personable. I've also collabed with them a couple of times. Yeah. Yeah. However, MHC is on my exam tomorrow. Good luck. I don't think I offered all that much in terms of testable ideas, but good luck on that. As humans, we're born essentially knowing nothing. Practically all we know can do are learned, and learning from masters who can teach is a great way to learn. Bring on the calm stuff. I would love to do it just for my own ego too, because it is so important to me that I get to feel good at it when I am teaching it, because it makes me feel competent, and I think that's worth doing as well. Stop labeling people. We don't have to have autism or ADHD or be passionate about one particular subject. Yeah, I agree. That's kind of like, wow, I'm like, I do think it's funny. I urge people not to, but people can't help it, right? Like if people who are neurodiverse identify with me because of certain traits that I have, that's just the connection between humans, and I just have to set the record straight that I don't fully share your experience of the world, that everybody has their own unique biology and psychology, and we're all a unique fingerprint on this earth. But there are certain aspects of my being that I think are relatable and not relatable, and that's just the normal part of being human. AI can't replace my thinking if I don't think. That's so true. Based. Surprise Pants is based. Haven't seen Avatar. Is good. It's really, really good. It is from a storytelling perspective, I think the most technically proficient cartoon ever. I don't know, is there a better one? And that's why I'm so careful. It's technically proficient. Many people will enjoy that. It's also beautiful. It's a beautiful show. Yeah. The aviation world recognized the over-reliance on automation and resulting decay of neurological capabilities issues years ago. Proper use of labor-saving tech so we don't get too weak is an issue. Yeah. There's like the whole, like, that's why I did this paper, right? Like I said from the outset that like this paper is a way for me to brush my teeth, to eat my veggies, to do my squats, do my push-ups, get some exercise in, so that I do not decay into just my comfort zone of picking only cell and molecular biology papers and not venturing out occasionally into other fields that challenge my understanding of how the world works. And I think that as long as you are actively always doing that, you shouldn't have a problem. But the thing about the path of least resistance is that, oh boy, is it frictionless to slide down the path of least resistance, right? Like it is so smooth and uncontroversial and easy to always do the thing that is easy. But we should strive to be better. Not that I do a great job on my own, right? Like, I say all of this not as like some wise guru that knows more, but as someone who is also victim of this happening, right? Like, I feel like I am worse at responding to messages. I am getting worse at paying attention to longer videos. I am choosing to forego engaging videos more often than I am like game slop, some analysis of some video game that I can put on in the background. Like, the reason why I feel so passionate about these topics is because of the effect it has on me personally, and just being like, the pot is boiling, and like, I don't think huge scale societal change is coming to save us, but just being aware and actively making attempts to reestablish some of the inconveniences of life for the sake of the quality of your being, I think is good. I don't think that's too controversial to say. All right. I think that has been a good and fun yap session. I think that is where I will probably call the stream. Thank you all for showing up. Thank you for spending some time with me. I had a good time talking about my favorite protein. Yeah, I think maybe the next stream might not be for like 2 weeks or something. I am going to be transitioning back into the hermit shell where I do nothing but work on my video all day and night. But at some point I would like to do like a rhetorical analysis of SciComm stream. And I will probably see you there. Stay tuned for some exciting stuff on your feeds, I think. All right. Bye, y'all. Goodbye. Bye-bye. Bye-bye.